Hi, everyone. Can you hear me? It is such a pleasure to see you all here. Thank you so much for coming out this morning. I'm Janine Cavender-Bears. I'm the new director of the Harvard University Herbaria, and I'm so excited to be celebrating 100 years of the Farlow collections with you all. What a wonderful collection that you're going to hear about today, you're going to get to see, and we're going to get to learn so much about fungi and cryptogams today. I'm so grateful to all the speakers for traveling from far and wide to come here to help celebrate today. And I'm delighted to be here. So what you are going to hear today is we're going to start off with Don Feaster, who has spent a career working in the Farlow, curating the Farlow, directing the herbaria, and he is going to take you through a history of the Farlow, and I'm super excited to hear that. And then David Hibbett is going to take us on a journey talking about the importance of biodiversity collections in general, but then the hunt, doing community science and fungal evolutionary biology on the hunt for the tiger sawgill. And then Brent Mischler is going to tell us about spatial phylogenetics and its application to bryophytes. I'm really excited to hear that. I missed your talk in Madrid, so I'm super excited. Terry Henkel came all the way from the very northern part of California to talk in the middle of his teaching to talk to us about 30 years of fungal research and collecting in the Guyana Shield. I guess I've known Terry most of my life, and as he's been a constant reminder about the importance of fungi in everything I do. And Kathy Aim is going to be talking to us about the innovative use of collections and the diversity and importance in rust fungi. And then finally we'll have Rebecca Yar, who came all the way from Scotland to talk about lichen diversity and using collections as conservation baselines. Well, I'm just really grateful to learn from you all today, and I'm going to give a few thoughts. I also want to point out that you're going to get to see, for those of you who signed up, you'll get to go on tours of the glass flowers. These are extraordinary collections with exhibit on pathogens. This is Jenny Brown, who curates the glass flowers. This is such a gem, and so I hope you enjoy that. I want to give a special thanks to Jeanette Everett. So as you know, I haven't been on this job very long, and Don pointed out to me that Farlow was turning 100, and so we cooked up this idea, and Jeanette really put this into action. It's just amazing everything that this group has done, and I also want to thank Michaela Schmull and Tracy Barbaro and Anthony Brock and all of you who've done so much behind the scenes to make this work. Okay, some things that I've just always found fascinating about fungi, and one is that they enabled plants to colonize land. This incredible partnership 470 million years ago that allowed plants to move, photosynthetic organisms to move from water to land, and that was a major revolution in life on Earth. And the fascinating things we can see in specimens like these fungal lumps in the earliest liverwort lineage, the Haplometriopsida, that go back some 420 million years ago, just documenting how important this relationship between plants and fungi has been. the incredible importance of fungi for the global carbon cycle. These partnerships are fundamental to the way that carbon moves through ecosystems. Huge amounts of carbon is drawn through plants into fungi, into the soil. So last summer I was at the Madrid International Botanical Congress. Some of you were there too, and Alex Antonelli, who kindly sent me his slides, was talking about this symposium of the state of the world's plants and fungi that they ran to discuss the findings and ways to fill knowledge gaps in plants and fungi. And from that came a whole body of work and a Q report documenting the gaps, documenting how far we've come in describing species and how far we have to go. It's estimated that there are 2.5 million species of fungi on Earth, so that's what people are thinking now based on that synthesis. And if you compare that to other organisms, it's a lot more than the number of plants on Earth or the number of animals that are vertebrates, although not as much as the invertebrates, 8.5 million. But the thing about the fungi are that 90% of fungi are yet to be discovered. That's a staggering number. So it is the group, and not just fungi, probably also other cryptogams. It's a staggering number of the world's biodiversity that we still don't understand. And, only 0.4% have even been assessed for their conservation status. So compare that to animals or plants, so animal invertebrates, plants, or animal vertebrates. 80% of animal vertebrates have been assessed for their conservation status, but very, very few fungi have. And here's a graph from Muller et al. Showing the accumulated number of species that are on the red list since 2003. So as more and more species are getting described, we're also learning that many of them are threatened. And so that's led to a series of efforts to try to recognize fungi as one of the three kingdoms of life critical to protecting and restoring Earth, and to really doing conservation for fungi and cryptogams in general. Okay, so one thing I can tell you from firsthand observations, I can tell you a little bit about why we have 90% of fungi and cryptogams that are not yet described, because I've watched my entire life how difficult this work is to do. Of course, we have all kinds of new methods to do this now, but collecting, painstakingly isolating, and describing under a microscope the species that are on planet Earth is really hard work. So this is me with my sisters back in the 1970s in an oak tree, and we watched this guy. This is my dad, Jim Cavender. He's emeritus at Ohio University, and he spent his whole career going around the globe collecting dictyostelids, these social amoebae, cellular slime molds that you can't really see with the naked eye, but you sort of can, and they have to be isolated. They've been the keys to understanding the evolution of multicellularity. They're fascinating organisms, but it's just really hard to find them, isolate them, and describe them. And so I watched that unfold growing up, although I didn't really understand what was going on for the first part of my life. We would go on these family vacations, and we'd always camp in the middle of nowhere, and my friends would get to stay in hotels and stuff, and we'd be off in the middle of nowhere camping all the time. And then we'd go on these cool hikes up the mountain, and I thought that was super fun. And then later I come to find, Dad was publishing the cellular slime molds of the Rocky Mountains. You know, this family vacation in Appalachia later turned out to be the cellular slime molds of the Southern Appalachians. You know, and there's my older sister in front, who's now in finance, and my mom in the back with my baby sister, who's now the director of the Huntington Botanical Garden, following along, and I'm there just trooping along, and that's how I spent my childhood, is following my dad around collecting cellular slime molds. And that was a ton of work. So he spent his life doing that, among other things. He did a lot of teaching, but so after a lifetime of collecting, isolating, describing species, we're now at about 200 dictyostelids. So of the 2.5 million species that are on planet Earth, 90% unknown. These are not even fungi. These are not true fungi. These are social amoebae. This is just the work it takes to describe species. And using molecular methods, we're going to go a lot faster. But this is just a ton of work. And the collections in the Farlow, the work, all the work that you all do is incredibly important. Well, they finally named so at the end of his career, they named a genus and a family after him, which is kind of fun. So I think that even though I am a plant biologist, I did grow up understanding the importance of the things underfoot. And this is a picture I drew when I was a kid of an oak tree, and somehow I must have been aware of everything below ground that was supporting these trees. And yeah, so that's probably 1978 that I drew that picture. I also grew up learning something about lichens. I spent 4 years in high school mapping lichens, monitoring them for their abundance and diversity in my hometown in Athens, Ohio, and did that for 4 years, using them as indicators of air pollution. This was based on studies that were going on in the United Kingdom at the time. So that's what I did in high school. And then, of course, my dad got all of us into helping him with stuff. So I was living in Germany. I'd been living in Germany on and off for multiple years before I started my PhD. And, you know, I wanted my parents to come over and visit. Well, Dad just couldn't just come over and visit. We had to do a whole project. So ended up driving my dad all over Germany, and we published this paper. We isolated and described the species in Germany. What's kind of funny is that if you look at the address, we published this while I was here as a graduate student. And like on the slide, I don't know what, Fakhri Bazaz was my professor, but we published this while I was a graduate student here all those years ago on the slime molds of Germany. And that was right after the unification. So this map that is a whole Germany was actually was amazing to draw at that time. But in the end, like, I rebelled. Like, I am not going to work on those tiny little things. I'm going to work on the biggest thing I can find. And so I spent the next 20 years working on oaks and have looked at them in so many different dimensions and have really appreciated being able to see them. But of course, I did always know that what was below ground was really critical to the success and the functioning of the oaks. And I did try during my postdoc and beyond to do the work to describe the mycorrhizae that were on these species and to do experimental work with them and to do all the microscope work. And it was just so difficult. And so I have enormous respect for all of you who do this work. I ended up giving up on that, and after morphotyping hundreds and hundreds of tips of ectomycorrhizal fungi on oaks, and then sequencing them, that was the way things were done then. I decided I was going to stick to above ground and let all of you do this work. So I just will watch and admire you. But I do know the importance of the interactions between land plants and mycorrhizae. So I'm going to go into mycorrhizae for a bit here. just because I know how important they are and I'm so interested in them. So these are just like cartoons showing the arbuscular mycorrhizae and ectomycorrhizae, two of the major types. And they function differently, and they associate with different plants. So on the left is a phylogeny showing tree of life of plants with the associations with ectomycorrhizae, and the rest are arbuscular mycorrhizae. I couldn't actually hear what you said.
The wrong slide. No.
No. Oops.
No, it was earlier.
Here's the
Oh, there it is.
there's the phylogeny. Sorry, I'll use the pointer. So you can see the ectomycorrhizae and the arbuscular mycorrhizae, gymnosperms and angiosperms. This is from this paper from Steidinger et al. And maybe we'll hear more from Terry later, but there's really different geographic distributions of ectomycorrhizae versus arbuscular mycorrhizae, although Terry's going to talk to us about the Guyana shield and what's going on with those monodominant forests there. And there are also really different nutrient and carbon dynamics with mycorrhizal types. So nitrogen is higher in arbuscular mycorrhizal plants across latitudes. And this is in green leaves, senescent leaves, and in roots. And and it's also, soil carbon is higher in communities dominated by ectomycorrhizal plants. So you get higher nitrogen in systems that are dominated by arbuscular mycorrhizae and higher carbon in systems dominated by ectomycorrhizae. So there's some fascinating things that are still not completely understood about why this is. That is the stuff of the ecological work that moves beyond the herbarium collections. I'm going to, in the last few slides, I'm going to leave you with questions that I have about the role of fungi in the oaks taking over North America. So some 40 million years ago, as the earth was cooling and drying, the oaks, which were in very northern latitudes, circumpolar region, probably came over across land bridges some 55 million years ago and were hanging out in northern latitudes. As the earth warmed and dried, the tropical taxa that were here died off, and the oaks started radiating throughout the Americas into Mexico, where they sped up their rates of diversification. But what's always been fascinating is that the white oak lineage in the blue and the red oak lineage in the red, and these are the live oaks, the Verentes, they diversified sympatrically and in parallel, and you can sort of see that in this cartoon. They started at high latitudes, they radiated east and west of the Rocky Mountains and diversified sympatrically and in parallel. And why is it that they were able to do that? This is actually from my PhD thesis many years ago in Florida, where you see different lineages co-occurring together in different ecosystems. So the scrub system, the sandhill system, the hammock system, and in each of those you find different lineages occurring together. And there's so many hypotheses for why you get this pattern of phylogenetic overdispersion. It does help explain why oaks are so abundant in North America, because they were able to diversify sympatrically and in parallel and were pre-adapted to the climate as it was changing to a temperate climate as that came in. As that climate appeared in North America, they were able to diversify and become the dominant lineage in North America. So what role did the fungi play? Well, this is interesting work that we've done from an applied perspective, looking at oak wilt, which, if you look at the two major lineages, These are the red oaks. These are the white oaks. The red oaks get infected by this fungus, Bretziella phagocerum, that spreads below ground between neighboring roots. And so trees, one red oak tree, and I'm using red oak in the sense of all the species in the Lobeti lineage, the roots will graft and the fungus spreads right from one tree to its neighbor. And within a year, it kills the tree. And this happens very fast. The immune systems in the red oaks are very ineffective. So this is a conduit, a vessel, and the fungus spreads through the vessel. The plant tries too late to put tyloses into the vessel to stop the movement of the fungus, but instead it ends up clogging up all the conduits and wilting. So it basically kills itself with its immune response. Whereas the white oaks have really got this figured out, and so they're able to be very precise in the way they put tyloses, where they secrete these gels that form tyloses that block the movement of the fungi. And so they can sequester the fungus and keep it from spreading. So they might lose a branch, but the whole tree doesn't die. So they also have different vessel diameters, which is probably part of this, really different immune systems. And so the red oaks, in general, are much more susceptible to fungal pathogens than the white oaks are. So they have the same symbionts, so they use the same ectomycorrhizal symbionts. There's a little bit of host specificity, but they also benefit from many of the same ectomycorrhizal symbionts, but they're very differentially susceptible to pathogens. We've done a lot of work trying to remotely sense oak wilt to help people manage it, because it can be managed, and we desperately are trying to keep it from coming here from the Midwest. It's spreading all throughout the central part of the continent, moving eastward, but it can be treated, and so if we can remotely sense it, we can help managers stop it. We've been working pretty hard on that. This is a really nice slide from Antonio Guzman, who's sitting there, showing how we develop remote models from remotely sensed satellite data to predict where the disease is. But let me get back to the pattern of overdispersion that I showed you from Florida. And when we've done analyses across the entire United States, using the forest inventory analysis data, we find the same pattern of phylogenetic overdispersion in all of the forest plots in the United States, although the traits themselves are clustered, meaning there's been you can read the papers, but the punchline is there's been convergent evolution in the traits, so they can occupy the same habitats, but it's different lineages, red and the white oaks and the live oaks, that are occurring together. That's the pattern of phylogenetic overdispersion. And so I would really love for somebody to figure this out who works on the fungi. Does the facilitation of red oaks by white oaks through mycorrhizal associations promote co-occurrence? So often white oaks are the first one to come in, and they have staying power. They're very resistant, and they're resistant both to things like drought stress, but also fungal pathogens. They bring in the mycorrhizae, the ectomycorrhizae, and they make a nice habitat for their red oak partners who can then come in and do well because the white oaks got there. But then because the red oaks tend to be faster growing, they might start to dominate, except that they're much more susceptible to pathogens. And so it could be this interplay between symbionts and pathogens that's really the fungi that are guiding this whole system and maintaining the co-occurrence of the oaks. That's just a hypothesis, but I would just love to know the answer to that and be able to test that in my lifetime. Okay, so the last thing I'm going to say about fungi is and cryptogams in general is they're just really cool. This is a picture of my little baby daughter some 23 years ago. I must have been tormenting her with cool fungi. And I hope that we can listen today with the curiosity and enjoyment to hear what everyone has done, because there's so much to know that none of us can know all of this, and we can learn from each other and engage with curiosity today. And I have signed on behalf of the Harvard University Herbaria, but you all can sign individually this 2030 Declaration on Scientific Plant and Fungal Collecting to use evidence-based collection strategies, strengthen local capacity, collaborate across taxa and disciplines, collect for the future, and share the benefits. So I hope today we can foster these conversations and clarify to the world why they are so important. I'll stop there. Thank you. Okay, Don is going to tell us about the history of the Farlow collections and who the person behind the Farlow collection was, the original collector. So thank you, Don.
Thank you, Janine. It's really great to hear about all these fungal topics. Mine is a tough job, 100 years in half an hour. And I have to admit, one of the things I did, I started a slideshow and I put it together and I thought, this will never work. So then I did a second slideshow. And I thought, maybe I could make this work. So you're seeing the second slideshow now, not the first. I want to point out as well that this is a meeting of the New England Botanical Society. And it's not just any meeting, it's the 1,181st meeting of the society. And this is appropriate. It's 125 years for the society. It's also true that William G. Farlow, our namesake here, was one of the big instigators of that organization. So it all fits together in a certain way. There is a guest book, which is back in the back somewhere, I think, and the society is collecting signatures, and they've been a wonderful sponsor for this event as well. So I'll point out also that today is All Saints' Day. It's also Day of the Dead. That may be very appropriate as we move forward. The other things of the year, we're not the only organization that is celebrating 100 years. The Morgan Library, where we have a little bit of a connection that I may mention. United Way is 100 years old this year. The Grand Ole Opry is 100 years this year. And the Boston Bruins joined the NHL 100 years ago. So big time here for the first 100th of the Farlow. So this is the reading room at the Farlow, and many of you know this. It's a wonderful space housing the collection. This is my text for today. David Leventhal in Heritage Crusade and the Spoils of History. As doctrine, heritage is mandatory. It comes to us willy-nilly and cannot be shed, however shaming it may be. To share a legacy is to belong to a family, a community, a race, a nation. What each inherits is in some measure unique, but common commitments bind us to others with our group. Whatever its burden, a heritage ought never to be denied. Self-respect requires us to accept, husband, and transmit it. So, what's our community, and what's the family here? Well, we're centered around cryptogamic botany. And I know I need to define this. These are spore-producing organisms, sometimes referred to just as plants, but they're much more. It's an archaic term. Goes back to Linnaeus, where we had two big groups of organisms, the phanerogams, those with flowers and seeds and so forth, and the cryptogams. And what organisms does the Farlow Reference Library of Cryptogamic Botany contain? Well, it's fungi, including lichens, it's algae, bryophytes, myxomycetes, even bacteria, but not ferns and fern allies. They have vascular tissue, and they're not included in that collection. Those are in the collections of the Gray Herbarium and the Arnold Arboretum. It was pointed out to me that we have some plants infected with viruses as well. So what do we have? We have 1.5 million specimens in 4 kingdoms under one roof: fungi, plantae, protists, monera. Now, I inserted this little quotation from Roland Thaxter, and I'll talk about Roland Thaxter later on, but I will read this except part of it. The popular reaction to the term, on the other hand, may be well illustrated by some of the superscriptions in the files of the Herbarium of Letters, which nonetheless have somehow found their way to their intended destination. And then you can see the variations here, ending in cryptogamic, as if such free and easy handling of its spelling, or even mild profanity, could make the term more respectable or more intelligible to the laity in the person of the postman. Now, for some of you who know about Roland Thaxter will realize this, but I will point out that's one sentence. And that's the way he wrote. So where do we begin with this family, with this community? Farlow grew up in Boston. He was from a wealthy family. His father owned railroads. He went to Harvard as an undergraduate. He went to the Harvard Medical School, and then he became the assistant to Asa Gray. Asa Gray, the North American botanist, who really put botany on an international footing in North America. Gray encouraged Farlow to study plants then including cryptogams, and that cryptogams should be his focus. Nobody else was doing this. This is a great area for Farlow to undertake. So some of the people in the lab love this photograph of Farlow, and I'll point out that he was a member of Hasty Pudding. And some of you in Cambridge know about Hasty Pudding. He played the piano, he was a musician, he was really wonderful in a number of different ways. So Farlow's big step to make himself a cryptogamic botanist was to go to Europe. And in Europe, he primarily studied with Anton de Bary. Anton de Bary studied ferns, he studied algae, he studied plant pathogens. He was responsible for working out the biology of the stem rust of wheat, the alternation of hosts. He worked on the potato blight organism, which isn't a fungus at all. In the Ainsworth History of Mycology, he's cited in every chapter, and those chapters are arranged topically. So Farlow went to study with him with the intent purpose of bringing back the techniques that de Bary was developing. At the same time, Farlow studied algae with the French phycologists and took up that topic. Back in Cambridge, Farlow started and worked on many projects. The algae of New England, the bibliographic index of the fungi of North America. He published an exocadi, these organized sets of specimens of algae. He worked on a host index of the plant pathogens for North America. And posthumously, the Icones farlowiana came out. Now, these are just major projects. They aren't his research papers. He did many, many research papers as well. I put one specimen packet here, and I'll refer to this later on as well. That specimen packet is Perinospora or Plasmopora viticola. That's the downy mildew of grape. When Farlow came back from Europe, he was at the Bussey Institution, which was kind of Harvard's agricultural college, and he was working on plant pathogens. Among those were these mildews, and he prophetically said, I'm studying this fungus in North America. Should it be introduced into Europe, it would be disastrous. It was, and it did. It was introduced. So he looked at this, he understood about the susceptibility of different varieties and so forth, and worked on that plant pathogen. And I'll come back to this a bit later on. So at Farlow's death in 1919, he left his library to Harvard with certain stipulations. It was to be housed in a fireproof building, was to be located in conjunction with the cryptogamic herbarium, all these collections that he had put together. The arrangements for the space, that is the approval of these plans, was put in the hands of Roland Thaxter. Roland Thaxter had been his student and colleague. And the gift, the library gift, was 10,000 volumes on cryptogamic topics and a collection of maybe 50,000, we're not quite sure. So Roland Thaxter, put in charge, he had been a professor at Harvard from the early 1890s. He had studied with Farlow and worked on many different organisms. We think of him primarily for his work on the labellobeniomycetes, or the labellobeniales. These are ectoparasites of insects. And I've shown you one here. Let's see if I can do this right. This is a drawing of his. They're fantastically illustrated monographs, whatever group he was working on. This happens to be Zodiomyces, one of these labellobeniomycetes that grows on water beetles. And this is a camera lucida drawing. He would ultimately make this into a final plate. We published a paper on this and titled the paper, Fireworks Under the Microscope. It's just such an exuberant fungus. But he worked on many different things. He was an artist. Here's from his sketchbook, sketching out one of these zygomycota. He collected many different fungi. Here's a myxomycete that he collected. Yes, we keep them in boxes. And we keep them in boxes for safety, not because we think they'll run away, but they're kept in the boxes. This was one of his monographs on the Endoganaceae and his little signature here, which is so characteristic. This is a cup fungus, Winia americana, that produces a big mass from which the apothecia rise. We did molecular work recently, not so recently, a while ago, showing that this is a combination of the tissue from the Winia, from the discomycete, and armillaria. And here's Thaxter in the lab, in the upstairs room at the Farlow. And here he is at Kittery Point with Myabe, one of Farlow's students. So what did Thaxter need to do? Well, he needed to approve a place for this collection to go. He needed to raise money, and that money was to support a renovation and addition. He needed to raise funds for the development of, or for the library and for a librarian. And he needed to increase the collection. And by the time he died in 1932, there were about 300,000 specimens in the Farlow. I can give 2 seminars on Thaxter. So what was the ultimate placement of the collection? Well, it was in the old Divinity School library at 20 Divinity. Here's the building from the front. Of course, all of this is gone. The HUH building attaches at this point. There's the cross. And then in order to house the collections with the library in this building, an addition was made here in the back. And one of the things you notice is that this is kind of setting out by itself. None of the buildings surrounding the herbaria were there yet. It's called one of Harvard's most picturesque buildings. I will say, yes, picturesque. We get flooded basements occasionally. There were periods when I came in on weekends when it was raining because the roof was leaking and I had to empty the buckets. It was very picturesque. These days, all of the collections are air conditioned, including the library, and that's been a big improvement of this. So here's the Rogues Gallery, the curators and directors over time. And you know about Farlow, or a little bit about Farlow now, and you know about Thaxter. When Farlow died in 1919, an assistant professor and curator was appointed, and that was Lincoln Ware Riddle. And you see Riddle there in the middle. Riddle was appointed in 1919. He'd been a professor at Wellesley, and he'd been a student with Farlow and Thaxter. Unfortunately, Riddle died, he was a lichenologist. He died within a year. He died in 1920, so very short term. Two people were appointed following Riddle, William H. Weston, Jr. and Carroll William Dodge. Both were appointed assistant professors. Dodge was also appointed assistant curator. Weston went on to teach for his entire career at Harvard. He died in 1978. Dodge did various things. He worked on various fungal groups. He worked on lichens. Some would say that was not the best part of his work. He also wrote the first textbook on medical mycology, and that was done at the instigation of Roland Thaxter. Thaxter knew that it should be done. So Dodge was responsible for moving the library, organizing the herbaria, managing the acquisitions that came into the collection during the 1920s and 30s, which was a great growth period. David Hunt Linder, followed Dodge in 1932. I always stop and say about Linder, he's our movie star director. I love his photographs. You know, there's something about them. Linder worked on many, many different types of fungi. He also published on diatoms, and he died in 1946 at a very young age. Many mycologists know Rolf Singer. Rolf Singer was at the Farlow for 7 years on fellowship and was acting curator after Linder died. His famous book, The Agaricales in Modern Taxonomy, was written while he was at the Farlow. We have one of his original drafts. William Lawrence White came to the Farlow in the 1930s. He studied at Cornell with the mycologists there. He worked on various of the cup fungi, and he was hired on to be a bibliographer. And I'll talk about him a bit later on. During World War II, he worked for the Quartermaster Corps. He was killed in a car accident in 1952. Elka Mackenzie, who published under the name of Ivan Mackenzie Lamb, joined the Farlow in 1953 and retired in 1972. And as I've been joking, the last 50 years of recent history, here it is. Okay. And one of the interesting things, thinking about community and family and knowing about people, these are the folks that I've met who have this connection. William H. Weston had been retired for many years by the time I came to Harvard in 1974. But he would come in, and one of the joys was that we used to have a parking lot behind the Farlow, and the curatorial assistants would see him parking his car and coming to the Farlow, and they would come running to me and say, "Cap Weston's on the way, Cap Weston's on the way." It was just, just real enthusiasm about him. Carol Dodge visited several times over my early years. He also came to the New England mycological group meetings, which was kind of a precursor of Mass Myco that's happening tomorrow. But he didn't drive, his wife Bertha drove, and she almost always got some kind of ticket or citation when she was driving to these activities. He gave us his herbarium, a large herbarium of Antarctic and other materials. And Singer and Elka Mackenzie, who I first met as a graduate student myself. So I put together this slide. It's a composite, a collage of many of the publications of the Farlow Herbarium. And these, I'm not going to go through all these, but they include the journal Farlowia, which was instituted by David Hunt Linder, I.M. Lamb's Lichen Index, the algae north of Cape Cod, Martin Zimmerman and I.M. Lamb, this New York Botanical Garden publication, which was by Geneva Sayer, one of my mentors, who came to the Farlow in her retirement from Russell Sage and organized the bryophyte collections. And so forth. And most of these would never be done without the resources that are offered by the herbarium and the library. So where are we today? There's about 1.5 million specimens, and I've listed out some of the important collections, but those in many ways are immaterial and it's incomplete. But I should say that the collection is very rich in type collections, these collections that tie names to the organism. And we have the library, those 10,000 annotated books that were Farlow's. He was a great bibliographer. He wrote in everything, and he talked about everything that, or he indicated what he knew about a particular book. But that's now about 70,000 items and includes an extensive library. So what about these specimens? Well, here's one just to give a little analysis here. So you see the original packet over here, and we can unpack that packet in various ways. There are labels in different handwritings. There's a bibliographic notation, Bull-Tor Bot Club, 1899. This fungus was first described as Cantharellus by Underwood. In 1910, it was placed in this genus Paleozelus. The Farlow annotation is here. You can see Farlow's handwriting. This is a good day. This was on a really good day. But what that says is rather Craterellus than Cantharellus. So he'd looked at it, he'd studied it, and he had an opinion about it. This is part of the original label, And this is an annotation by Timothy Baroni from 1980, and that was when Tim was here as a postdoc in the collection. Now you're all looking at this. That's the specimen. And you look at that and you say, how can anybody get anything out of this? Well, we can. And what we can do is that these can be rehydrated, they can be sectioned, we can look at the anatomy of the organism, we can look at the spores, we can gather all kinds of information from these dried specimens. And most of our 1.5 million specimens are dried. There are a few that are in liquid, but most of them are dried. So, thinking of specimens again and some of these personalities, during World War II, the Quartermaster Corps instituted a program to look at tropical deterioration. That is, how fungi in particular were affecting substrates of various kinds. So it could be wood, it could be canvas, it could be a shoelace, it could be the shoe itself. But these deteriorating items were cultured, or the fungus was cultured from them, identified, and recorded. And these four were part of that project. This is William Lawrence White. He headed up the project. And David Linder, who did much of the identification of the fungi that came through. William Weston consulted and worked on the kind of biology of these fungi. And he was responsible for freeze-drying fungus cultures. That is, putting them into a state of stasis so that you could come back to these years later and revive those cultures and work on them. And after his death, when materials from his home were being brought in, a number of these ampules of dehydrated, freeze-dried cultures were brought in. We know that at least one of them was viable because we sacrificed it to see if it would grow out. But what they did, they grew the fungi, and these are all specimens that are in the collection. This is a dried fungus culture, so we can go back here, we could do morphology, we could look at that, we could probably try some DNA from this, we haven't tried it. They photographed them, and they drew and illustrated them as part of their identification. So here's this vast trove of fungi from around the world, a lot of it tropical world. This one isolated from a plastic canteen from New Guinea. So here it is, a snapshot that we have of organisms really from around the world that were collected for this very specific purpose. This is Elso Barghorn. Elso Barghorn was a paleobotanist. He was here at Harvard. He did his PhD here at Harvard and worked on plant anatomy as a PhD student. He became interested in wood rot. What's happening? What organisms are involved with it? So he was part of this project in the sense that he had a kind of sub-part of it. He took blocks of wood and put them out in the environment, and then would go back and look at the wood and analyze what was going on. He, Barghorn, and Linder also did the first real comprehensive study of marine ascomycetes. And they brought together the knowledge of the time, and then they added collections that particularly Barghorn had made. So a very influential piece of work. Now I know that there's interest here in Guyana, so I wanted to point out this particular set of specimens. In 1924, Linder went to British Guiana, as it was called then. And did a collecting trip. He collected a lot of flowering plants and ferns, but he also collected many, many fungi. And this is the same specimen. I've shown it in two different ways. So this is a Daldenia, a little black ball-shaped fungus. And you can see from the first over here, he called it one thing. He went back later on and re-identified it, and here it is here. The specimens that are databased or barcoded, here's the barcode for that. And this specimen was used later on to do a chemical study, and this was done in the lab of Mark Stadler, who's in Germany. And he's very interested in the compounds that are found in certain fungi. In this family, the Xylariaceae is a real target for his work. But you'll see it's annotated chemically, which is really interesting information. But you'll also see that they didn't quite read the label correctly. BG, not British Columbia, but British Guiana. And I point this out because this is one of those cases where it's really important to know the collector, know the expedition, and maybe even know the curator who could help you with that. So anyway, that example. Now, I'm really taken by the work that Farlow did in 1912, looking at the chestnut blight. And many of you know the story of the chestnut blight. We know now that the organism was introduced, the fungus was introduced from Asia. It was introduced around the port of New York. It moved very quickly from New York and basically decimated the forests of chestnuts. We still have chestnuts in the forests, but they're shrubs. They never or rarely get to be mature. So this was really a big issue at the turn of the century and following. And Farlow did this study. It was published in Science in 1912. And I wanted to show one of the specimens that he used in the study. So here's the specimen, here's the label, and it's part of one of these exocadi, multiple sets of specimens that exist in different institutions. And this one is from an Italian collection here. So Farlow studied this. He made slides. Here's these little glass mica slides. Here's a little more slide. Here's another one that he sent off to other folks to look at. But his goal was to try to figure out, now, what this had been described, it had been described by Merrill, as you can see, as Diaporthe parasitica. Farlow began looking, using his collections, using what he had in his own collection. He began studying this, and he realized a couple things. One was that it wasn't a Diaporthe at all, and he put it in Endothea parasitica, which is where I learned it as a student. Later, this has become Cryphonectria parasitica. So here's a study, and it's very interesting because in one of Weston's publications about Farlow, he talks about Farlow bringing him into the lab and saying, would you look at the microscope here? I've got these slides. I want you to compare them. And Weston was a student in 1912. He was a PhD student. Weston looked at the slides and he said, well, I think they're different. And Farlow said, some people would disagree, and he walked away. And they in fact were different. He was using Weston to check himself. But what's interesting in this is that there's a passage, and it's not all here, but to produce dramatic effects on our native species, which have not, through long exposure, become more or less immune to the fungus. So he's talking about the fact that this is introduced and the plant on it's infected is going to react differently, perhaps more violently, than the plant that it was native to. So this harks back to his work, early work on the Plasmoptera viticola, the downy mildew. So there's a modern counterpart here that I wanted to mention. We're on oaks. We're on oaks today, so I thought we'd just continue on oaks. This is a paper that we have NSF support to work on powdery mildews, and this is primarily being done by Michael Bradshaw, who's now at the University of North Carolina State University. And this is one of the papers in that powdery mildew series. You can see there's a phylogeny. You're not going to read that phylogeny, but there's the phylogeny. But realize within that phylogeny now, we have a type specimen. And that type specimen had been overlooked at the Farlow. And there's the type specimen. Who would have thought to look in a North American collection for a specimen from France? But there it is. It's a tribute to how specimens got exchanged and moved around. With having that specimen, we now know and can assign reliably a name for this powdery mildew. And this powdery mildew is important because it's a North American powdery mildew on North American oaks that was introduced into Europe. And of course, in Europe, it's a much more, on those oaks, it's a much more severe infection. So here it is. We can kind of mirror the 1912 study with looking at this and thinking about how we verify, how we know about the names and can study these invasion processes. In the middle here, just to make sure you know, the powdery mildews, because they produce masses of conidia, masses of spores on the surface of the leaf. Here it is. And late in the season, you get these structures, which are called chasmothecia, in which asci are produced. So here's an ascus. They have beautiful appendages. So beautiful fungi. Here's an interesting story. So just a couple more things here. I want to mention the Friends of the Farlow. The Friends of the Farlow, Geraldine Kay, who was the librarian at the Farlow, and I established this in, I can't believe it, 1983. We've already missed an anniversary. And we did this to call attention to the collections and their value and to promote the collections. We produced a newsletter. We support an annual lecture. This would be it. This was the weekend. We always did that. We provided funding for graduate students from afar. So we would support students who wanted to come and use the collections. And funny kind of aside, I once was at an MSA meeting and I sat down at a table and realized that every person at the table had had a Farlow fellowship. It was wonderful. It was wonderful. We raised an endowment. We established the Harvey Pofscher Fund that supports mid-career investigators to come and work. And there's an annual foray, Clara Cummings lichen walk that Elizabeth Kneiper has been good at. And here's some friends to thank. And a particular friend is Bob Edgar, who curated the the diatom collection for many years. This is only part of the diatom collection. They're all on slides. And I'll point out that David Linder worked on some of these diatoms from the Boylston Street archaeology site. But where are the women? And here they are. The primary woman of the Farlow was Lillian Horsford Farlow, who supported Farlow, but after his death was the major support, financial and emotional, for Roland Thaxter. She gave many dollars to the collection. Gertrude Riddle was the widow of Lincoln Ware Riddle. Her handwriting, sample here, is everywhere. It's all through the collection. She did these labels. Hilda Harris was librarian. Florence Lovejoy was a librarian, and my favorite quote from Florence Lovejoy is, "The cards in this file are in the order in which they were found, which is no order at all." Sylvia Earle, marine biologist, supported the phycology collection. Geneva Sayre, my mentor, worked on the bryophyte collections, on literature, and was just a wonderful person. And Brent would say the same thing. Geraldine Kay was librarian. Judy Warnement, also librarian. And Michaela Schmuel is back at the back, and she's the newcomer. So Emerson wrote on attending his college jubilee, "Cambridge at any time is full of ghosts. But that day, the anointed eye saw the crowd of spirits that mingled with the procession in the vacant spaces, year by year as the classes proceeded, and then the far longer train of ghosts that followed the company. The long winding train reaching back into eternity." So I haven't mentioned all of our students from Farlow's to mine. I haven't mentioned all of the curatorial assistants. I haven't mentioned visitors. I haven't mentioned lots of things. But I wanted to end with this because I think it sets the stage for both the past and the future. And then at night in the library, the ghosts have voices. Thank you.
Thank you, Don, for that wonderful history. And we're going to start now with the first of our invited speakers. And I would like to welcome David Hibbett. He's professor of biology at Clark University, and he received a BS in botany from the University of Massachusetts Amherst, a PhD in botany from Duke University. He did postdoctoral fellowships at the Totori Mycological Institute in Japan and at the Harvard University Herbaria, and I think that's when I first met you, David. He joined the faculty of Clark University in 1999, and he'll be talking to us today on the importance of biodiversity collections, professional networks, and community science in fungal evolutionary biology, or hunting the tiger sawgill. Thanks, David. Welcome.
Okay, thank you, Janine. I apologize for making you read that title. If I could have my slide, please. Or do I have to do that? Nope, that's not it. Okay, there we go. It's a tremendous honor to be part of this celebration of the Farlow Herbarium's centennial. The Farlow is hallowed ground for all fungal biologists, as for the reasons that I think Don just showed you. And for me personally, it's really sweet to be here because I was a postdoc here for a number of years in the 1990s, and that was just an amazing time. I learned so much during that period, and I received such incredible support from the HUH community, including the staff in the magnificent botany libraries, and I will forever be grateful to the institution for that continuing support. Okay, so here's the title, and after I submitted this, I realized that This is a terrible title. This is the kind of title that I tell my students never to write. It's long, it's wordy, it's vague, and it doesn't tell you what the talk's about. So let me tell you what the talk's about. So the talk is about evolutionary innovation. It's about the origin of novel forms in the history of life, specifically in fungi, and it focuses on this organism here. This is Lentinus tigrinus, more commonly known as the tiger sawgill. And I guess I'm advancing myself. Okay. And here's the outline of the talk. So I'm going to tell you a little bit about my motivation, give you some background information, then more than you ever thought you could want to know about Lentinus tigrinus, then a little bit on new materials and new results, and then I'll close with some speculations on the maintenance of the polymorphism. And now you're all asking yourself, what polymorphism? Well, I will explain. And I will issue a little caveat at the outset here, which is that I don't have a complete story to tell you, okay? I've only been working on this problem for about 30 years, so we're just getting started. So if you expect a complete story, I'm afraid I'm going to disappoint you. Okay, if you talk to a lot of mycologists, they will tell you that there was one fungus that drew them in to the field. Maybe not the one they've worked on their whole careers, but there was one organism that was their gateway mushroom. And for me, it was this. This is Hericium erinaceus, also called the lion's mane. And I first found this when I was a college student at UMass Amherst. I didn't know anything about mushrooms at that time. And I was completely blown away when I found this thing in the woods. I just couldn't believe that there was something so weird and beautiful. And I brought it to Howard Bigelow, and he was completely unimpressed. But nonetheless, I was hooked. And years later, many years later, I learned that that fungus is actually closely related to this coral fungus here, as well as this crust fungus, and also this gilled mushroom. And now I was really intrigued, because how do you get the transitions between all of these diverse forms? How has this incredible diversity come to exist? And there's so many questions around that topic that have kind of obsessed me for my career. You know, what's the historical pattern of transitions between forms? What are the genetic mechanisms that allow developmental programs to shift so that you can get this diversity? What are the ecological conditions that promote or allow these new forms to arise? And what are the evolutionary consequences of these new forms? Are some key innovations that spur diversification? Are others evolutionary dead ends? And so on. So those are the kinds of questions that I've been thinking about, and will actually be addressing to a certain extent through this talk on Lentinus tigrinus. So keep all of that in your head as we go through this material today. Now, I need to give you a little more background information before we can get into Lentinus. All of these fungi here are what are called hymenomycetes. So hymenomycetes are fungi that produce their spores on the surface, on the outside of the fruiting body, whether on teeth or coral tips or gills or on crusts or whatever. And hymenomycetes, although they're incredibly varied in gross morphology, in basidiomycetes anyway, they share this mechanism of spore discharge called ballistospore. And it's this incredibly sophisticated, cool, precise discharge mechanism. The physics have been worked out, some work here at Harvard and elsewhere. So this is a shared conserved feature of hymenomycetes. And you can contrast hymenomycetes to gastromycetes. Gastromycetes, which literally means stomach fungi, are fungi that produce their spores on the inside of the fruiting body. So they include puffballs and things like that. So they form their spores internally, and they've lost ballistospores. So on the far left is the basidium, the spore-producing cell of a hymenomycete. The three on the right are various gastroid basidia, and they're really weird because these things don't do ballistospore anymore, and the basidia have gotten strange. We also know that gastromycetes have evolved many, many times. So this is a very big tree. I'm only going to show you 3 slides with phylogenetic trees. You'll be happy to hear. This is a tree of 8,400 species that my postdoc Marisol Sanchez Garcia put together. The red branches are the gastromycetes. Some of those branches are very small, 1 or a few species, so we put dots around the outside to show you where those gastroid lineages live. So gastromycetes have evolved many, many, many times, and they've yielded some really strange forms. So you have bird's nest fungi and earth balls and truffles, cannonball fungi. These are all gastromycetes, and stinkhorns. everybody loves stinkhorns. So this transition to a gastroid form has yielded a lot of evolutionary innovation, amazing forms, and how does that happen? That's a problem that I'm interested in. And it's really hard to figure that out because, we have a pretty good idea of the phylogenetic placements of these gastroid lineages now through phylogenetics. We know what their closest relatives are in many cases. But gastromycetes are so divergent morphologically from their hymenomycete precursors that it's really hard to reconstruct how the transitions happen. So like scleroderma on the right, the pigskin earthball, I think is the common name, is closely related to Gyroporus on the left, but at the macro and micro scales, they really have nothing in common. So it's hard to figure out how those transitions might occur. So there's a group of fungi called secotioid fungi, which are gastromycetes, that are interesting in this regard because these are forms that are morphologically really similar looking to their hymenomycete precursors. And in the olden days, they all got their own genera. Now that we're placing them into lineages with their hymenomycete relatives, they've been transferred into hymenomycete genera. So like Nevatogastrium on the far left there is now a Foliota. Thaxterogaster, named for you-know-who, on the lower left, has been transferred into Cortinarius, and so on. But these are really interesting fungi because they're gastromycetes, but you can look at them and say, aha, yeah, I see how this is derived from a hymenomycete. And this is a schematic from a famous paper by Tom Bruns in 1989 that kind of encapsulates one view of how this happens. So on the far left is a bolete, a hymenomycete, and the idea is that there could be some macromutation, the first red asterisk, that gives you an enclosed fruiting body. And that's the secotioid form in the middle there, intermediate. And then, if that form is lucky enough to pass through the filter of natural selection, then sort of classical Darwinian gradual evolution, refinement of the gastroid form, loss of ballistosporia and so on can take place, the two asterisks, leading in this case to a false truffle. So this is kind of the dominant paradigm right now for evolution of gastromycetes. One big macromutation that gives you a secotioid form leading to this thing that then is elaborated. So now we'll get to Tigrinus, and I'm going to start with a little history, talk about morphology and genetics, and then we'll put it in a broader context of the genus. Here it is. This is the type illustration. There's no type specimen for the species. This is from Bouillard, 1782. This picture, by the way, I've never handled this book, but I was able to get this image in high quality through the wonderful Biodiversity Heritage Library, which has been an important resource for my work. Anyway, it was Agaricus tigrinus, the tiger agaric, because back in the day, everything with gills went into Agaricus. The great Swedish mycologist Elias Fries transferred it into his new genus Lentinus. So Lentinus tigrinus is a Friesian name. This is 1825. This is all Europe, right? So Europe at the time, that was where the cutting edge of science was happening. But then, in the middle of the 19th century, a voice in the wilderness, Thomas Lee, who was a lawyer and botanist in Ohio, finds a strange mushroom, and he sends it to the Reverend Miles Joseph Berkeley in England, because that's what you did back then. You corresponded with the great experts in Europe, and you sent them material. Berkeley was at Kew, so he was sitting at like the spider at the center of the imperial web, just drawing in the riches of the empire to study and describe and name. Nothing wrong with that. And Berkeley writes that this thing looks like it might be a new genus, but I am convinced that it is merely a very curious but monstrous state of our European species. And I may be too sensitive, but I find that just slightly offensive. And I don't know if it's the merely or the monstrous or the our species, but I detect a whiff of transatlantic chauvinism. Although Berkeley actually he loved Lee, and he named a beautiful mushroom called Mycena leana after Lee. So I should go easier on Berkeley. Anyway, here's the monster. So it's just after Halloween, so you're all ready for monsters. I think it's actually kind of cute. But what you can see here is that this doesn't have gills, right? This has an enclosed hymenophore. This is a secotioid fungus, right? It doesn't release spores directly into the air. They're trapped inside the fruiting body. So this is the secotioid form of Lentinus tigrinus. Berkeley said it's, all the things that he said. But then in 1895, another American mycologist, Andrew Price Morgan, sort of threw down the gauntlet in a taxonomic sense. And he says, not only is this a different species, it's a different genus, and he calls it Lentodium squamulosum. So that's 1895, around the time that a lot was happening here. So Lentinus tigrinus, at least in North America, is one species with two forms. It's polymorphic. It has a secotioid and an agaricoid form. And I want you to just reflect for a minute on the fact that the sole function of a mushroom is to discharge spores. That's its only job, right? The one on the left, the secotioid form, traps its spores inside the fruiting body. The one on the right does what all hymenomycetes do. So this form has to have huge consequences for spore discharge, which should have significant fitness effects as well. So bear that in mind. Here's another picture, some fresher material, slightly prettier picture. So one of the wonderful things about Lentinus tigrinus is that you can grow it in culture. And that is not true of many gastromycetes. We haven't figured out how to do that, but this thing grows like crazy on sawdust medium. So in my first postdoc, before I came to Harvard, I was at the Tottori Mycological Institute in Japan, where they really know how to grow mushrooms. And I did a study with Shigeyuki Murakami and Akihiko Tsuneda on this species. And we used SEM to look at development. So this is a very young fruiting body. You can see the margin of the cap. You can see those ridges, right? Those are the young lamellae, the young gills just starting to form, and they're going to get broader and taller as the thing goes on. What you're looking at here is you're sort of looking face on two gills. So those are two gills coming down. You're sort of looking toward the stipe. And what I think you can see is that at the margin of the gills there, some of the hyphae are starting to grow out. They're proliferating from the edge of the gill. And then at maturity, they completely enclose the gill. So in this one, you can still see all those gills, right? In others, in more extreme forms, the gills get very contorted. But here you can really see how this develops. And this is what it looks like in a macro photo. Agaricoid on the left, secotioid on the right. And because we can grow this thing in culture, we can get single spore isolates, which are haploid, and we can do crosses, and we can grow up those mushrooms. We were able to do some basic genetics. And what we found is that this morphology exhibits simple Mendelian inheritance as a recessive gene. So it looks like a single gene. And I'm putting gene in quotes because we have no clue what that locus is. It could be a protein coding gene that has a mutation that changes its shape. It could be a single base pair change in some transcription factor binding site, or my favorite hypothesis, it could be a non-recombining supergene. So we have no idea what the genetic basis of this form is, but we know it exhibits simple inheritance, and we are working on this. But I don't have data to present today on genetics, but it's a major goal of the lab. Okay, so that's an introduction to tigrinus. I want to broaden this out a little bit now, give you a little bit of a sense of the geographic range of this species, and look at some of the relatives of this organism. So in the olden days, like in 1994, if you wanted to understand the diversity of a group, of a species, where it lives, what it is, you had to go to herbarium materials, and you had to write to curators and ask, would you please be so kind as to send me these materials so that I can look at them? And so that's what we did, and they did. So we got data from 71 specimens, including here at Harvard, and we drew this map showing the distribution of the secotioid form in eastern North America. So that was wonderful, but it's, that's not a trivial undertaking to do all those, to get all those loans. Now we have wonderful informatics resources. So this is the Myco portal, the Mycology Collections portal, which lets you go in and look at the holdings at many, many herbaria, including Harvard, of course. And you can see there are 1,480 records of Lentinus tigrinus in the Myco portal, and that's fantastic. Some of them even have images. And the images usually look like this. And And this is cool. This tells us who collected it and when and where. And there's some metadata. It's in low wet woods. That'll become important. But what does it look like? I mean, one difference, there's been a huge effort to digitize herbarium holdings, which is fantastic. But with organisms that are in packets or boxes, high throughput digitizing is really, really hard because you have to open the packets. But we have other resources as well now. So this is a page from iNaturalist, which has 4,550 observations of Lentinus tigrinus. And I have an automatic feed to get an email update every day when new materials are deposited of this group. And I've just stopped looking because they're just coming in so fast. And the records give you a lot of information. Like they give you habitat, they give you, even if they don't talk about habitat, the pictures often show. It's like this one from, I think this is Texas. You can see that this mushroom is fruiting right above water. Unfortunately, usually there's no collection associated, but still, there's a lot of information, useful information. So this is a map from the mycoportal. This is the Lentinus tigrinus map. Let me get this straight. The circles are collections, so those are herbarium specimens. The triangles are observations. And what this shows me, I think this is wonderful. This really shows how these community science observations and herbarium collections have really been integrated into our understanding of the distribution of diversity, with the understanding that both kinds of records have their limitations. But community science records are really important these days. And here's the map from INAT, and a quote above from David Pegler, who did the monograph on this species, on this genus, Lentinus. He says it's been reported all over the world. Misidentifications are frequent, and he says which organisms are most often confused with it, and says it is essentially a north temperate fungus. So the big blobs in North America, Central America, Europe, and Asia, that's probably real tigrinus. And the others are probably misidentification. So I'm going to show you some pictures of other species of Lentinus, starting with things that are not particularly closely related to tigrinus, that are in different sections, and then other things that are in a section that Pegler called Tigrini, which contains tigrinus and close relatives. So here's Lentinus crinitis. This is not in that section, Tigrini. Here is Lentinus sager-keju, also in a different section, section rigidi. This is Lentinus squarocilis. This is an INAT photo. This one is very often confused with tigrinus, and I think you can see why. And now here are some things in the Tigrini that are really hard for me to tell from tigrinus. This is Lentinus retinervus. This is one of only 2 records of this species on INAT, so this is a very rarely observed and rarely collected species. This is Lentinus glabratus. There's only 1 observation on INAT, and this is it from the Cayman Islands. Note that it's growing out of water. I think this is also Lentinus glabratus. This is one of my collections from Florida. I was skipping out on the MSA meeting that afternoon when I went out and found this fungus. And then here's Lentinus concinus from Australia. This is the only record of concinus on INAT. So this is another rarely collected species. The type specimen is here at Harvard. And so what we've learned from looking at these online records, we've learned a lot about where these organisms are, and importantly, we've gotten this sense that the section Tigrini, those mushrooms are associated with water. It's not just tigrinus. So here's tigrinus from England, from the First Nature site, and the commenter notes that they're frequently in these low wet woods. A little more information this is a summary of data from INAT that my students Thomas Rail and Sophie Irons assembled. So here is the Secodioid form in green dots in eastern North America, but not in the very deep south, at least not very often, not in Florida. The Secodioid form of this fungus is only recorded from North America. It's not in Europe. And in 2011, Ed Grand published a phylogeny, so this is phylogeny number 2, showing with just a single gene that the Eurasian tigrinus and the North American tigrinus form 2 sister lineages. So looking like maybe two species. They also did some crossing studies and found that they can mate, at least they can mate as far as forming dicaryons when you mate 2 monokaryons. And they said, you know, looks like two species, but they still mate, so they didn't do anything nomenclaturally. There are, however, also some morphological characters that separate the European and North American things. I mean, first of all, the Secodioid form is only in North America. The European things also have a partial veil, which you can see in this fruiting body that we fruited in the lab from material from Germany. And that partial veil is not observed in our North American species, as Berkeley might say. Okay, so that's all background. What I want to do now is just give you a sense of what we've been doing recently, the really unfinished stuff. So I'll talk about some of the new material we've got, and I'll show you our preliminary phylogeny and some taxonomic thoughts. So for the work that we want to do, we need population samples because we want to look at selection. We want to do some population genetics. And so we've got a couple populations here in Massachusetts, one up in Topsfield, the Ipswich River, and one down in Concord on the Concord River. The Concord River population is in the Great Meadows National Wildlife Refuge, and Larry Millman, who's sitting right there, found this population for us and brought us to it. The Fish and Wildlife Service declined to give us a permit to collect Lentinus tigrinus at Great Meadows, but it being the hometown of Henry David Thoreau and all, we thought that some civil disobedience might be warranted. So here is Devin Rose Lever and Thea Henry from my lab surreptitiously collecting mushrooms at Great Meadows. We don't go there anymore. But you can see it's right on the banks of the Concord River on roots and logs of silver maples. That's its preferred substrate here. We also go to the Ipswich River. The Mass Audubon Society has been a fantastic partner. They did give us permission to go and collect there and let us use their canoes. So that's how we access the site there, is we take canoes. So here's Christina Martin and Iris Knowles, two other undergrads collecting at Ipswich River. Here's Thomas Rail, who I believe is here, who's my PhD student, also collecting. So you see these mushrooms are right in the water. So that's the Massachusetts populations. We also have a population in Illinois that we found through INAT. We were looking for places where there's a lot of collections and that are accessible. So we decided to go out to Illinois to collect on the Chicago River. And we had wonderful support from the Illinois Mycological Association, which is a mycology club of mycophiles in the Chicago area. The clubs are really important to the conduct of science these days. And one group that I've worked with a lot locally is the Boston Mycological Club, which has been just great. And I know that there are a bunch of BMC people here. And BMC also has a historical connection of sorts with the Farlow. Anyway, so we went to Chicago, and we got great support. This is Rocky Hotby from the IMA, and you can see he's risking life and limb to help us collect tigrinus and not fall into the Chicago River. Here's Thomas Rail performing kind of a acrobatic sloth-like maneuver, seeking out this mushroom. I mean, this is not skipping through the forest with a basket collecting porcini. Okay, this is hardcore field taxonomy. And here's Thomas and a visiting student, Ryuka Izuka, in the Little Tykes laboratory in the basement of the Airbnb where we stayed making collections or making isolations. We also went to the Field Museum, to the herbarium there. The Field's another institution that Rolf Singer has a historical association with, and because herbarium samples are also tremendously important for our work. Here's Alexander Bradshaw, who's a postdoc in the lab, sampling a small amount of material from a specimen. We take pictures and we can provide these back to the herbarium so that they can put them on mycoportal, so these collections become more valuable in that regard. We also rely on our network of collaborators and colleagues. Terry Henkel, who's in the back, has been tremendously helpful. This is not Terry. This is one of his field assistants on the Ireng River in Guyana. You'll hear much more about Guyana, but I got in touch with Terry because I know he goes to interesting places, and I asked if he'd look for these tigrinoids, and I said, well, you should look along riverbanks on wood and rivers, and he gets to his sites using water, and they found it. And they found some really cool stuff. So this might be Lentinus retinervis. This might be Lentinus glabratus. This, he called it gray shaggy, and maybe someday we'll translate gray shaggy into Latin. But super grateful for those materials. And so here's the last phylogeny slide. We generated ITS data, sort of the barcode locus, put those into a big data set of previously deposited sequences, over 400 samples. So the tree that on the far left is all of Lentinus, including those things from the other sections. The pullouts are parts of section Tigrini. Okay, so we've got group A, which I'm calling Lentinus sensulato, 30 isolates from the United States, and then some things from the Philippines and Russia that we're still trying to figure out. B is Lentinus sensu stricto, so this is the clade that the name Lentinus tigrinus will have to go with, Europe, Eurasia, et cetera. C is probably Lentinus consinus, or at least it might be. We just have ITS on that other people deposited. D is probably glabratus. E contains that retinervis from Guyana and gray shaggy. The red things are all of our new materials, and the tree on the right is a genome tree. Okay, so the tree on the left is just ITS, which is this small fragment of ribosomal RNA genes that we use as the barcode. The tree on the right is whole genome data. All the things in red are new. These are very fresh results. I think these trees are, I think this is about 72 hours old. So this is the first time I'm presenting any of this. But the genome tree really upholds what the ITS tree suggests. Groups A and B really look like strongly separated different lineages. They have morphological differences. They have geographic signal. They are different species. So what do we do with them? We're going to need a new name for clade B. Pegler, who did the monograph, put a bunch of names in synonymy with tigrinus. Two of those were from North America. One is Lentinus ravenellii, and the other is Lentodium squamulosum. You already heard me talk about squamulosum. So a little bit about on these names. Lentinus ravenellii was a name by Berkeley and Curtis, his American collaborator, from 1849. Squamulosum, you already heard about, by Morgan, 1895. That's a picture of Ravenel. So Ravenel did not name the fungus Ravenellii. It's considered very bad form to name a species after yourself. But Berkeley named it for Ravenel. And by the rules of botanical nomenclature, priority is really sacrosanct. So the oldest name is the name. So the oldest name, by a lot, is Lentinus ravenellii. But I have a problem with that name. And it's because Ravenel was a great American botanist. He was from South Carolina. He was a planter. He was also an owner of enslaved persons. And he was a really outspoken defender of the institution of slavery. And I find that distasteful. And I would prefer not to honor Ravenel with the name of my favorite mushroom. But the code of nomenclature does not allow you to reject an older name because it's distasteful. That's something that some people think maybe we should change, but maybe it'll change someday. Fortunately, there are other criteria that we can bring to bear on this issue, in particular, name usage. So here's from the Google Ngram viewer. I put in Lentinus ravenellii, Lentodium squamulosum, Lentinus tigrinus, and this is mapped against the entire corpus of Google literature. The red line is tigrinus, the blue line is squamulosum, and thankfully, no Ngrams found for Lentinus ravenellii. So this name has just been ignored. It is not in the literature, and we can argue on the basis of providing connectivity to older literature that squamulosum should be the name. So I anticipate that we will someday be writing a paper proposing Lentinus squamulosus conserved against Ravenellii on the grounds of its common usage. Okay, so all of that, which I hope you found interesting, that's the easy part. People in my lab may not want to hear me say that, but this has been the easy part. We've basically confirmed Ed Grand's single gene tree. We've basically come up with firm support for the idea that there are two species. But there are other questions that are, and that's not a trivial accomplishment, that's a big deal, I think. But there are other questions that are really motivating to me, like the genetic basis of this transformation and the population genetics. How is selection acting on this new form? Why do we have this polymorphism? And we don't really have data to answer that question, but an imaginative scientist is never constrained by an absence of data. So I want to close with some speculations about why we still have this polymorphism in North America. As I was saying, the sole function of a mushroom is to discharge spores, and these two different forms have got to have wildly different spore dispersal properties, right? So you would think that one would probably be superior to the other in terms of natural selection and should drive the other one to extinction. But we don't see that. We see this maintained polymorphism. You can find both of these forms side by side in populations. And why is that? I think it has to do with the relationship of this fungus to water. So this organism really likes to fruit right at the edge of water in logs and on roots that are partially submerged. So you can see the algae here on the log. And here's another collection. This is from the Ipswich River. The circled, yellow circle, that's a primordium. So you can see that baby Tigrinus is just coming up. So as the waters recede, the mushrooms come out. Great. But the problem, of course, is that waters don't just recede, right? Waters and rivers can come up. And mushrooms can become submerged. So here's a collection with one above the water and one below. Here's the picture from the title slide. This is a semi-aquatic mushroom. It's not fully terrestrial, it's not fully aquatic, it's semi-aquatic. And it inhabits this environment in which you have these stochastic fluctuations in water level within and across years. So this is 3 years on the Ipswich River. The light blue shaded parts are the fruiting season. This thing fruits from early May well into October. And you can see the water level changes dramatically. 2022 was a drought year. You mushroom hunters will remember that was a bad time. 2023, we had tremendous amounts of rain. River was going up and down like crazy. And here, 2024, it's been kind of a mixed bag. But these fungi are fruiting all year long during the season. And the changes in water level are stochastic. They're unpredictable. And so I think, and this is my hypothesis, that this results in what's called balancing selection. So a form of natural selection that maintains two different forms, two different genotypes, and that it's related to the fact that they occur in these fluctuating environmental regimes. I suspect that the secotioid form has superior performance when it's submerged, that somehow spore dispersal works better for those mushrooms when they're underwater as compared to the guild forms. But the typical hymenophyceae form might have superior performance when they're exposed in the air. And that is pure speculation, but I think it's reasonable. And we have a tiny bit of data. This is from master's work by Sophie Irons in my lab, and she's found in our Ipswich River population a slight preponderance, a slight but statistically significant excess of heterokaryons, so individuals that carry the gene for the secotioid form and for the agarikoid form, so a slight excess of those. No evidence of any assortative mating or anything like that. And when we get our population samples from this site and from the Illinois site, maybe Concord, we'll hopefully be able to take a better crack at that. So that's the Lentinus story. That's the story I have for you today. Beyond anything specific to Lentinus, I hope I've made the point, or I hope I've illustrated the kinds of resources that we're drawing on now in systematics and evolutionary biology. Herbarium material has always been, will always be incredibly important. We have all these great tools like MycoPortal to find things. The citizen science, community science community is generating huge volumes of really important data that we must not ignore, must figure out how to integrate into our taxonomic data streams. And if we can put all of these resources together, I think that in the years to come, our understanding of the diversity of life is going to become much, much richer than it already is today. Okay, Lots of people have helped. I've mentioned some of them along the way, but others have helped. The NSF is supporting our work. These other organizations I've already mentioned. And I don't know if we are doing questions. Are we?
You have 1 minute for questions.
Thank you. Terry.
David, about the cryptogams form, those forms still have normal ballistоспoric form basidia?
Yeah, so the question from Terry Henkel was, does the Cocodioid tigrinus still have ballistospory? And the answer is that it does. So it looks like a relatively recent evolutionary innovation. And I mean, so let's come back in 10 million years and see if it's lost ballistospory and evolved into a full-blown gastromycete. Good question. Any more? Okay, well, thank you very much.
During the coffee break, you can ask David your remaining questions. I'm going to introduce Brent Mischler now. He will be speaking about spatial phylogenetics with applications to bryophytes. And Brent comes to us from UC Berkeley, where he's Distinguished Professor Emeritus, Curator of Bryophytes at UC Berkeley in the Jepson Herbaria. So Brent spent 6 years in the Farlow Herbarium while he was a PhD student at Harvard, and then he was a professor at Duke for 9 years, and then he spent 31 years at Berkeley. He recently retired and is now Distinguished Professor Emeritus there. His research interests are in the ecology and evolutionary biology of bryophytes, as well as the theory of phylogenetic systematics, and I'm really excited about this talk on spatial phylogenetics. And so, without further ado, Brent, thank you.
Thanks very much, Janine. Can everybody hear me okay? Hope we don't get feedback. It's really great to be here. Thanks for having me. I have some slides here somewhere. Are mine just right? So I actually was here, as Janine said, for 6 years, and I realized in putting this together, makes me feel old, but I just barely missed the 50th anniversary. It just happened. Don, you said you came in 74, and that would have been the 50th. So, you know, I've been associated with the Farlow for a while, and it was a great time, and I want to thank everybody for that time, and especially Don, who provided me great space for a graduate student. I had Toby will vouch for this. I had, you know, a big office, and I also got to have Geneva Sayer as an office mate, who was a big influence on me too. So it was a great time here. And what I want to talk about, I guess I'm the token plant person here. Fungi are really cool, but there are some other organisms studied in the Farlow Herbarium. So I'm glad we at least had one token. But there's great bryophyte collections and also great algae collections as well. So I'm not going to talk about species today, particularly other than this slide. It's been a interest of mine all the way since those days. But, so I don't want to really argue about it. My radical view now is I don't think the species level is useful or interesting at all. But for the purpose of this talk, we don't need to go there. Let's just say whatever they are, Dave was giving us a really good example just now. Whatever they are, they're not the same thing across the tree of life. So if they have some meaning in particular groups, that meaning is different in different groups. So really what unites the study of biodiversity and lets us compare things on the map around the world and across different groups is the phylogeny. They all have a history, and the different lineages that are on the tree are different to some extent or another extent from each other. And the point of what I'm going to be talking about here is to not count species or use species for any purpose in studying biodiversity or conserving it, but rather using the tree itself as a measuring device. we're interested in endemism, which is the restriction of some lineages to small parts of the Earth's surface. That's particularly interesting for conservation, of course, but it's also interesting for trying to get at evolutionary and ecological processes. So the range size of lineages is an important thing. And that's also not just species. So even if species are a level, there's levels bigger and smaller than them, and they all have distributions, and they're relevant to study of process. So the bottom line for what I'm talking about here is to use the whole phylogeny as far as we have it, as far as we understand it, to do things, to do academic things like studying ecology and evolution, and then also to do conservation and practical things like that. So the key measure, some of you know this, but I'll just review it here just in case. I'm going to show just a few slides to review the main concepts. The traditional metric of biodiversity and endemism would be counting how many species are in a place and then what's their range size. This illustrates, these two phylogenies illustrate that the same number of species, 14 in this case, they could be closely related to each other, or they could be distantly related to each other. If the 14 species are closely related to each other, there's actually less diversity there than in the case where they're scattered all over the tree. So the key measure here is called phylogenetic diversity. It's been around for about 20 years. It just connects the dots. So you take the terminal taxa, the species or whatever they are, in a place, and you put them on a phylogeny, and you add up the branch links that connect them. And that's the measure of diversity. That's called phylogenetic diversity. And then there are modifications of that. One of them is phylogenetic endemism, which is just looking at the range size of each branch. One common way to do that, phylogenetic endemism metric, takes the regular PD metric and divides it by its range size. So if it has a little tiny range, that stays big. If it has a really wide range, that gets small, and then you measure PD with that, and it emphasizes the rare lineages. This is enabled by some of the things that Dave was just talking about, the advances that have been made in digitizing herbarium collections and in gathering metadata from other sources, from literature, citizen science, the tremendous increase of data in GenBank. It's possible to make a phylogeny of like 50% of the plants in North America just from GenBank, which we've done. Then the ability to make big phylogenies. Since I was a graduate student, when it was hard to make a phylogeny of 12 things, now it's quite easy to make phylogenies that are, maybe not perfect, but are well estimated from thousands and tens of thousands of things. So what I'm talking about, this field of spatial phylogenetics, was really only enabled within the last decade by advances in those three areas. We thought about it beforehand, but we couldn't do anything about it because we didn't have the data. So it has these advantages listed here on the left, and one of them is that since it's not using taxonomic rank, it gets you out of these debates about whether are these two species, or are they one, or are they two subspecies. For this purpose, it doesn't matter. What matters is how closely related they are. So it's relatively robust to some of these splitting, lumping arguments. And the basic idea is we're going to take the phylogeny and put it onto a map. We're going to have pixels of various sizes depending on how rich your data is and how robust your knowledge of geography is. And then ascribe what part of the tree is in each of those pixels. So you're essentially turning the tree into, if you've heard of GIS, Geographic Information Science, it's taking data and putting it onto maps and correlating it together. So we have GIS layers of many social, humanities sorts of data, but also biological data, environmental data, and we can begin to relate in a way that was inconceivable even 10 years ago, these different factors together. So just I'm going to run through this with some cartoon diagrams just to make sure it's absolutely clear. Here's a cartoon diagram of 5 terminal taxa that are related in this way, and the topology of the tree is the same, but the selection of 3 taxa that are in any one place is different. So we usually measure PD as a percent of the length of the tree rather than absolute number of mutations. And the reason you do that is to make it more comparable, because different genes evolve at a different rate. So you can compare 2 genes much easier if you're just talking about the percent of the tree that a branch occupies. So if you pick 3 there that have kind of short branches, the phylogenetic diversity is 55 or 55%. And if you pick them with really long branches, there's about 90%. You can't get to 100% with this selection of taxa because you can't cover the whole tree. So does this seem sort of clear? Your choice of which taxa are present in an area affects how much of the phylogeny is in that area. And then for PE, just to be sure we're clear on that, this is the same tree. The big red numbers there is how many pixels that terminal taxon is in. So there's a couple of widespread ones, and then there are 3 widespread ones and 2 narrow ones. And we divide the branch length, and it would look something like this. That one branch that's only in 1 pixel is really big, and the others have shrunk down, and if you enlarge them, they'd look something like this. And then we make a measurement just like we did on PD, except we use this topology instead. And it's in contrasting those 2 that you get some interesting information. So PE is just PD on this modified tree. Then there's a couple of more derived matrices that I'm going to use, and I want to just explain what those are. We talked about one modification of PD. Another one is the good old-fashioned cladogram C down there, where all the branches are equal, compared to the original tree. So the original tree with the original estimated branch lengths is A, and C is our artificially modified version of it, where every branch is equal. It's actually interesting to look at the ratio of A to C, because if you have an area with long branches, it'll stand out, because they're going to have a lot of above average. It'd be sort of like taking height measurements of people and then looking for concentrations, maybe in the basketball gym where there's unusually tall people or something like that. So we can find conglomerations or gatherings of long branches or short branches. And then we can also do that with, so if we range weight each of those trees in the way I described in PE and look at the ratio of those, it lets you see, particularly useful for conservation purposes, where the rare long branches are and the rare short branches are. You put them on a map and look at them. So we take our original tree, same cartoon tree, and we're comparing it with this artificial comparison tree to see where there's big branches and where there's little branches. And we've applied this to many groups, and even though I'm a bryologist fundamentally, we started out with all kinds of other groups. So I've worked on butterflies and angiosperms because they had better data. We finally felt we had enough data for bryophytes, and we gave it a try. So I'm going to show you that. But I just want to point out that it's interesting that RPD metric in the far left there, this is all the flowering plants in North America. This is this big tree, probably 50, 60% of them. There was a line almost drawn with a ruler down the middle of the United States. And you can pick up the fact there's a much older flora in the east and a much younger flora with lots of radiations in the west, just because there's a overabundance of long branches in the east and a overabundance of short branches in the west. Chile is also divided that way. Long branches in the south and short branches in the north. The locations where there are a lot of short branches are these centers of origination. And places where there's conglomeration of long branches often is what has been called a refugium, where you just have a lot of taxa whose climate has come and gone or something. There's some reason why they're hunkering down in some place. Okay, but now bryophytes. I really wanted to talk about bryophytes here. There's some wonder. That first of that mushroom you showed that really got your interest and got you into mycology, was that picture taken in California? The one you showed? Because I could tell from the moss behind it is a West Coast endemic. So even though you were talking about Eastern US, I said that has to be a, you know, when you're a botanist, you can never really appreciate like movies and stuff where, you know, this is supposed to be set in Africa and you're going like, no, come on, that's Southern California, I can tell. Okay, so bryophytes. There aren't as many as there are fungi, at least by the named amount. They're little relatives of the other land plants. I'll show you a simplified phylogeny in a minute. They contain 3 big groups, which you can see here, hornworts in the bottom right, and liverworts and mosses. Still controversial as to how closely related they are to each other. The green plants, which includes a lot of the algae as well that are also in the Farlow. The green plants is one of the big clades of life. At the bottom of that, that's about 1 billion years old or so. There are 2 huge branches of the green plants, one of which has the marine seaweeds, like you know, codium and things like that, ulva, sea lettuce. It has Chlamydomonas. And then there's some of the green algae, not a lot of them, but some of the green algae are more closely related to the land plants. Those are called the streptophyte green algae. And then we have the land plants. That's the topology of the bryophytes that I think is right, but there are a number of other ones out there. So that's one of these things. There was an early radiation of land plants, and it's pretty difficult to try to say what the relationships of the bryophyte lineages are to each other and to the vascular plants. So we decided to make an analysis of North American mosses with trepidation because the number of records is pretty low, even with all these sources of data. There was, after we cleaned the data, there was only about 746,000 records, which might sound high, but the North American study we did had millions of angiosperms. And so give it a try. We were able to represent about 2/3 of the known flora. So the results I'm presenting on here are based on 2/3 of the known flora and the phylogeny that connects them. So it's, you know, there's still some of the terminals are missing. And I have to talk about the spatial randomization. The phylogenetic diversity, which is, let's see, just regular species richness is A, and phylogenetic diversity is B. And you see how closely similar those maps are. That's because PD is an additive measure, and every time you add a species, you're adding a branch, and PD is going up. So there's an expected relationship of richness and PD. If organisms didn't care who they live with, it was just kind of random how they were associated, if this sort of, you know, neutral theory of ecology was true, and if the communities in any one place are just a certain number of taxa who kind of fumbled together, then there should be a perfect correlation of richness and PD. So the interesting thing is when the PD is more or less than you'd expect with that number of taxa co-occurring. Janine showed the example with the oaks of phylogenetic clustering versus phylogenetic overdispersion. You detect that this way. Phylogenetic clustering is when the organisms living together are surprisingly closely related to each other. Overdispersion means that they're too distantly related to each other to have come together by chance. So we have to use a spatial randomization really to test these patterns. Otherwise, they're just not that interesting. So what you do is you take the occurrences of the organisms off the map and you put them back on the map with 2 constraints. One is that the number of taxa that goes into a grid cell is constant, and the range size of each of the terminals is constant. And other than those 2 things, you do this over and over again. You measure PD, RPD, and all of these things. And you're looking for the outliers. We're trying to find the special places on the map that don't behave as expected by the neutral theory. So the PD randomization is the one that tells you what Janine was looking for, which is the structure of relatives living together or not. So significantly low PD indicates, which is red in this map, indicates close relatives living together. And on the other hand, the blue indicates significantly distant relatives living together. When you look on a really broad geographic scale, PD is always significantly low. Just because if you look at a whole continent, there's almost no taxon that doesn't care about the different environments between the tundra and tropical forest or something. So it is on a continent scale for angiosperms, the map is all red. So angiosperms are at that scale. But it is interesting that with the bryophytes in the southeast, which is where the older flora is of tropical origin, is overdispersed. The RPD randomization, I explained RPD is comparing the PD on the original tree with that comparison tree that has equal branch lengths, really shows the southeast as having really long branches as compared to the rest, except for just the Pacific coast out here, out in Terry's region. So we think that PD randomization has more to do with ecology, how the organisms are interacting, where the RPD may have more to do with longer term patterns of evolution and biogeography. So areas where there's a number of recently diverging groups are going to tend to be significantly low in RPD versus these refugial kinds of places. And I was talking to several people this morning about applying this method to different data. I will say that I'm talking about a continent scale here, but there's no reason why you'd have to do this. This could be done on just one valley, one river valley, and you were just interested in what's the distribution of biodiversity on that valley or that mountain. And I want to talk about canopy. We were hungry when we named this one. I'm getting hungry again, so it's good timing. This uses the RPE metric, which is also comparing the PE on the original tree with the PE on the comparison tree. It's a little bit elaborate. I'll show you my cartoon diagram of how it works. But we're trying to find what are called centers of endemism first. A center of endemism is a place where there's a statistically significant number of range-restricted taxa. And that's something systematists and biogeographers have been interested in for a long time, and conservationists, is finding those special places where there's lots of narrow range taxa. So the first step of this is doing that. And then once you have those, we're going to classify them into three, not equal categories, but three categories, one of which is dominated by rare short branches, one's dominated by rare long branches, and the other one is not dominated by either. It's still a center endemism, but it has elements of both. You've probably heard these terms before. They're fairly old terms. They originated in the California literature actually a long time ago. A paleoendemic is a lineage which is really old and has a small range. So a coast redwood would be one of those. And a neoendemic would be lineage which has a small range, but it's really new, with the presumption being the new one has not really had a chance to spread out yet, and the old one is probably maybe used to have a wider range, and at least now it has a narrow range. And the way it works is we have the original tree. That's the same cartoon tree we were looking at before. And then we talked about range weighting it. And then we have our comparison tree lower left. And if we range weight the comparison tree, then we're going to compare this tree and this one. And remember, I contrived it so there were two range restricted branches on that cartoon tree. And this one is shorter here than here, and this one's longer here than here. So you're looking for cases like that. You're looking for cases where the range-restricted branch is either much longer than expected or shorter than expected. Just look at the moss one here for a minute. We had no detectable neoendemism centers in this study. It's the first time we ever done like that had none. But in the southeast there and in Texas, you have these concentrations of range-restricted long branches. And then you have the mixed and superendemism kind of on the west coast. And it's strikingly different. I put the other two maps in just for comparison. Those are two studies done on the same scale. There's butterflies and the flowering plant one I already mentioned. We had originally thought the flowering plant one and the butterfly one ought to be pretty similar because butterflies and flowering plants are related to each other's evolution to some extent, but they were totally different. The endemism at that scale in the angiosperms is kind of all down in the south, whereas in butterflies it's in the arid southwest and in the west, and the mosses it's in the southeast. So there's different processes, different time scales, working on different groups. And certainly within any one of those groups, if you took a particular group of butterflies or a particular group of angiosperms or a particular group of mosses, they would have a different pattern themselves. This is what I talked about at the IBC. This is my talk that I gave there. Both depend on phylogenetic scale and they depend on spatial scale. The results that you get depend on how big a region you're looking at once, what your pixel cell size is. So it's sensitive to that. And it's also sensitive to how much of the tree do you look at once. Do you look at one little group of fungi? Do you look at all fungi? Do you look at all living things? And you're going to see different patterns, but that's okay because that's what you would expect, is that different groups have different patterns, and so you're going to see maybe a summary pattern overall, but you still want to dissect it and look at separate groups. In this paper, we also looked at sphagnum as a distinct clade. I'm not going to show that, but sphagnum had its own little deal. Okay, then finally I want to talk, so far what I've been talking about is alpha diversity, and alpha diversity is how much biodiversity is in one place. There's another big component that makes up the total biodiversity, which is beta diversity, which is how much difference is there between places. And there's a lot of reasons why you want to study that. One of them is to make regionalizations of an area and try to say what are the biotic regions of an area. It has uses in conservation when you're trying to compare a site you may be wanting to conserve with sites that are already conserved. And you probably had these metrics in ecology class. They're very common in species to compare how similar two places are in species. You have equations like these, and it's really simple. In the phylogenetic turnover metrics, you just substitute, instead of species that are shared and not shared, it's just what branch length is shared and not shared. So same equation, but you put in a different variable. So you can have your original tree. There's our cartoon tree again. You can have the range-weighted one, and you can do turnover with either of those. But we found empirically that the range-weighted tree is really good to do turnover with for doing biotic regionalizations because it's emphasizing the range-restricted taxa, which tends to be what your goal is when you're trying to say how many floristic regions are there in North America of a certain type. But you can use either one. So used on the moss data, so that thing on the left there is not a phylogeny, that's just a cluster diagram showing similarity in the phylogeny. So it's not a phylogeny, but its metric that it's using is similarity in shared phylogeny. It has a number of groupings in it that are color-coded, and some of them are called out as to where they fit on the map. And then, of course, you can go deeper in the cluster analysis, and you could break apart some of these areas. But these were the kind of primary clusters that we found in this paper, and what we are proposing as sort of the bryophyte regions of North America. Here's kind of put together on one map on the left. Those would be the bryophyte-defined floristic regions, which are not exactly the same as the ones you would get with angiosperms for sure. And then you can do things like looking at what environmental factors are influencing that. So you can take your information about phylogenetic similarity and you can compare it to climatic similarity and try to see what environmental variables are most closely correlated with the turnover, what might be defining these regions. The tool that you use for all of this is a software that's called Biodiverse. If you just type that in Google, it'll come right up. My colleague Sean Laffin from Australia does this, and it has both regular species metrics. If you don't want to give those up, it'll do all the species metrics too, everything, but it'll do all the phylogenetic stuff that I've been talking about here. And I highly recommend it. It also has a really good graphical interface. It's almost my last slide. Has a really good graphical interface, really good for teaching. When you get one of these diagrams like this up on a computer screen in Biodiverse, you can just sort of click on the clusters and light up the map and see where they are. So it's really good for like exploring your data and checking things out. And this is my last slide. I've written a very short review recently, which was 2022 or 23. So in the Journal of Biogeography, you can check it out here. It's open access, so you can see it. And I've summarized these and other studies. And hope I've saved some time. Hope for some questions and comments and things from people. So thank you very much. Yes, sir.
Really interesting that I'm wondering about the spatial pattern that you describe. Hi, I'm wondering about a spatial pattern that you describe, and you say that many metrics are sensitive to the pixel size. How you describe the optimal pixel size to do this study? And then all the environments share the same pixel size? Or do you think there's a distinction between?
Oh, I see what you're saying. What we've done is use the same pixel size throughout for ease of comparison. And I don't know of any theoretical way to do it, but in the study we did on California vascular plants, we illustrate the process. What we did was we took different pixel sizes and we looked at the results you get at those. And when you do like 5 kilometer squares, you get, it looks like just white noise. When you do 10 kilometer squares, it looks like white noise. When you did 15, you got a nice pattern. And then when you did 20 and 25 and 30, you got the same pattern. So it sort of emerges, and that doesn't mean, like for the bryophytes, we did 50 kilometer squares. For the angiosperms of California, we did 15 kilometer squares, and it all depends on how thick your data is. There is one good measure in Biodiverse that is the most useful one for that, which is called redundancy, which is how many records do you have of each taxon in each grid cell. So it's very bad to only have one record of each. Let's say you have 7 taxa recorded from a grid cell. If there's only one specimen each, that's not good. You would like to have more. Yes, Janine.
Okay, related and super picky detail. When you were explaining the phylogenetic endemism and you gave the cartoon example of how many pixels each taxon was in, are you also taking into account how far apart those pixels are? Or can you just like gloss over that? Or is it really just the number of pixels?
Yes, so what Janine just asked was, when we do PE, are we taking into account the distance between the pixels or just how many they're in? And the answer is, right now, how many they're in. Now, there's all kinds of modifications. This is in its infancy. Oh, and the randomization that we use is a little bit unrealistic because that also assumes, like I said, when you take the occurrences off and put them back on, it assumes they could be anywhere in the area. So it's unstructured. So that could be kind of unrealistic for all of North America because the taiga taxa are being, tried out everywhere. So there's also randomization methods in biodiverse that you can sector it if you want. It just, I'm nervous about how, since I want to use the methods to produce biotic regions, I don't want to start out assuming what they are and then, just randomizing within those. But no, there are randomizations that use distance, that when you put the specimen back, it has to be X number of kilometers from where it was, that sort of thing. So I encourage the young students in here that are just getting started to work on this stuff. The methodology is brand new, and there's a lot more that can be done. Yes, ma'am.
This is kind of mundane, but I saw that you showed that west of Mississippi, there was one thing. Oh, west of the Mississippi, there was one thing going on, and east of the Mississippi, in one of your maps. And I was wondering if there are any theories about how that works, because where the Mississippi starts, it's not this enormous thing, but the branches just happen so high up on the map in the upper Midwest. I was just curious about that.
Well, it was a striking result. We were blown away. We thought there was some problem because it's not really along the Mississippi. It's along a particular line of longitude. And what it is, it's probably environmental. So it's right at the place where the deciduous forest gives way to grasslands and deserts. And it makes perfect sense in retrospect when you look at it, because the groups, now that was angiosperms, not bryophytes didn't show anything like that. But with the angiosperms, these groups that have kind of hyper-diversified tend to be west of that line. Whereas the groups, they're very few by comparison to the Great Plains and Arizona and California and Oregon. There are very few groups in the east that have these kind of recent spectacular radiations. So they're longer branch taxa. Anybody else? I think now one more.
Yeah, so just hopping off of that, do you think there could be any interplay of other species or other large taxa that come into that? Because you have your kind of older lineages on the East Coast. You also had some in the Pacific Northwest, but you would have had like large megafauna coming in over the ice bridges that are known to be ecological engineers.
Could be.
Do you think it's possible that it was more a expansion of that due to these other organisms, not necessarily associated with trees or bryophytes?
Yeah, that's a really good point. So what he's saying is, it could be interactions with other taxa that are causing these things instead of just environmental. And it's a really good point to keep in mind that these patterns are only patterns. They're not processes. You have to infer process. So what we would say is that it's very interesting, but you would need to look more carefully to try to explain the process. So yeah, I think that could be, and thinking about these fungi, it could very well be where they're pathogens, that could set boundaries.
Okay, so in an effort to stay on time, let's go to the coffee break. Thank you so much.
Yeah, you're welcome.
30 years of fungal collecting in the Central Guyana Shield. What have we learned? And I think I recognize that person. Okay, so Terry is professor at California State Polytechnic University, Humboldt. And he received his Bachelor of Science in Botany from Ohio University, which is where I met him many years ago, a Master's of Science in Botany from the University of Wyoming, and a PhD in Botany from Duke University. His research investigates macrofungal biodiversity and ecological relationships in the remote tropical rainforests of South America's Guyana Shield and the Guineo-Congolian region of Central Africa. And Terry, just as a side note, has been a super athlete, rugged botanist doing things that are really incredible, and I'm just so excited to hear what you have to say.
Thanks for that introduction, Janine. And thanks, Don, Jeanette, and everybody for putting on this event. I have been to the Farlow Herbarium before. I was here for about a week in the late 2000s, I think, working with Matt Smith on a particular fungus in Don Feaster's lab. And I gave a talk then, too, not in this room. That person that's on this slide, I'll just name drop her right now. That is Mei-Lin Chen. She's been my partner in crime pretty much for all of this 30 years of field work that's been primarily in Guyana. I've got too many people to mention as far as acknowledgement, so I'll throw up a slide and point out some key individuals whose work or other contributions to this work are going to appear in the talk at various points. First of all, under the systematist list, Kathy Aim, who is here today, who's speaking after me. So I'm really going to try hard to run way over time to eat into Kathy's talk. Just kidding. Couple photographers, Todd Elliott and Noah Siegel. Both of those guys are pretty much world-class mushroom photographers. Noah's from Massachusetts, by the way. Some of you may know those individuals who I've taken on numerous expeditions to various parts of the tropics to get good field photographs of mushrooms. Matt Smith, too, among the molecular ecologists, who has been critical in numerous below-ground studies that we've done, environmental sequencing, and many grad students and postdocs along the way. But a little bit of origin story for me and how I really got started working in the Guyana Shield region has to do with really after I finished my master's, somehow I stumbled into a job with the Smithsonian Institution's Department of Botany, having no prior experience really in tropical botany. I got my master's at the University of Wyoming, after all, and was hired by Dr. Vicki Funk, who a few years ago unfortunately passed away, who was the Asteraceae curator at the Smithsonian and also the director of the Biological Diversity of the Guyana Shield Program, which the Smithsonian ran for about 25 to 30 years. Vicki hired me to go down to Guyana as a plant collector. One of the things you had to do when you were hiring on as the Smithsonian resident collector in Guyana is you had to go to DC and do like a 1-month intern at the museum, Department of Botany. So what did they do to the collector during that 1 month? You went around to the different curators who were experts in different groups, and they told you what they wanted you to find in Guyana. So the main charge was go to the most remote parts of the interior of Guyana and collect fertile plant specimens, press them, and send them back to us so we can examine them, look at them, and describe new species and develop the floristics archiving for the flora of Guyana. So John Werdak, been deceased for a while. John Werdak was the Melastoma curator at the Smithsonian. Werdak really sat me down and gave me marching orders of what to do. John Werdak was Bassett McGuire's right-hand man in the 1940s, 50s, and into the 60s on their great New York Botanical Garden expeditions to the Tepuis of Venezuela. So Werdak gave me these marching orders. This has got a video in it, I hope it plays. Henkel, when you get down to Guyana, don't concentrate on collecting on the summits of the Tepuis. We've done a ton of that already. The slopes are the terra incognita, the flora on the slopes of these sandstone mountains. And also, you must find McGuireanthus ioncanii. So let's see if this video plays. I'm not really good at embedding video files, so it's supposed
What
to.
we opportunistically just saw at our rest spot on this trip was this wonderful plant. This is McGuireanthus ioncanii in the Melastoma taceae. This plant is one of the rarest melastomes and poorly collected melastomes in the world. It was first collected by Bassett McGuire from the New York Botanical Garden in the 1950s for one of the early expeditions to Ionagana Mountain and hadn't been collected since. It's a single species in the genus McGuireanthus, monotypic. And when I was sent down here as a plant collector for the Smithsonian, John Werdak, who's a melastome specialist at Smithsonian, specifically told me to look for this plant if we did expeditions in the Ionagana region. So Bruce Hoffman and I in 1992 found this plant and made some good herbarium specimens, and that was the second record for this plant in history. But one of the key features is this sort of T-shaped flat inflorescence, and you can see right here that this plant is actually in bloom. There's one flower on that terminal appendage of the inflorescence. And so it has this flat, very sort of cross-shaped inflorescence, which is really weird for a melastome. It's a sprawling, semi-herbaceous plant, and it has the trinervy characteristic of the Melastoma taceae, but not very pronounced. And actually, there's more than 2 secondaries from the midrib, which is a little unusual for a melastome. So there you have it, an opportunistic sighting of McGuireanthus ioncanii in the Pacoima Mountains on the 9th of July, 2006.
And I saw it.
Over and out.
That was back before the days where video cameras on phones and stuff were very high tech. But needless to say, John Werdak was really pleased when we came up with a bunch of sheets of McGuireanthus ioncanii and sent them back to him. So the first thing I want to do is talk about trees in the South American lowland tropics. And with the neotropics, I'm constantly making the distinction because the tropics are defined by latitudes, degrees of latitude either side of the equator. And the fungi that we study, these ectomycorrhizal fungi, there's a distinct difference between montane plant communities that occur in Central America, the Andean highlands that are still in tropical latitudes, and really what we find in the lowland part of the neotropics, which are primarily in the greater Amazon basin. But let's talk trees in the South American lowland tropics. The ATDN is the Amazon Tree Diversity Network. As of 2024, we're up to 2,046 botanically inventoried 1 hectare plots for woody plants above 10 centimeters DBH. So this database has been growing since the early 2000s when Hans ter Stege, who's in the Netherlands, began putting it together. And he coordinates the data. This is from one of Hans's recent papers just showing the array of plots spread across the greater Amazon basin and the physiographic regions basically indicated by these acronyms, and this would be the Guiana Shield. So we've got pretty good distribution of the plot arrays. There are certain black box areas I want to point out because this will come into play later, like between western Guyana and western Venezuela, eastern Colombia. In the Guiana Shield region, there's a dearth of plots and therefore a dearth of information about plant diversity and or tree diversity, et cetera, in that black box. So now I have to clarify the multiple Guianas, because everybody goes, wait a minute, Guyana, Guyana, what's going on? That has to do with a lot of history and how this name has been applied. So Guyana was the spelling that was originally used for the non-Latin colonies in South America, which were British Guiana, Dutch Guiana, and French Guiana. Guyana is the name that was chosen by the Guyanese people when they obtained independence from Britain in 1966. Suriname, they didn't even do the Guyana thing, and Suriname, which was Dutch Guiana, has its own name. And of course, French Guiana is still a department of France. But in Spanish-speaking countries, namely Venezuela, the sandstone overlay that's on top of the Guiana Shield granitic bedrock, they call that region Guyana. And biogeographers that have been studying distribution of organisms in this elevated sandstone terrain, the Tepui country, call that the Guyana Highlands. So there's all your Guianas, but I've got colors coding the basic regions. And then the Amazon basin itself, which is pretty much the main drainage catchment of the bulk of the waters that flow through the Amazon system. So one of the early papers, well, first of all, so in the Amazon basin, you know, you look at these plots that are distributed in the ATDN database, you know, 200 or more, in some cases a lot more, tree species will be co-occurring in 1 hectare of forest. 1 hectare, that's 2.2 acres. That's typical for the central Amazon, and then it goes off the scale in the northwestern or upper Amazon here, where 300 species or more per hectare is typical. But the Guiana Shield region, I don't have it on the slide, but you mensurate a typical mixed forest with no clear dominant species in it. You have about 100 to maybe 120 species in a hectare, which is still really diverse by temperate standards, but also some trends in the floristic composition and life history traits of canopy tree species that really shift when you go from the basin up toward the Guiana Shield. Some of those that Hans pointed out in this early paper from 2006 was, you look at average seed mass of canopy trees, there's a trend towards increasing in the Guiana Shield. Wood density goes way up across families of canopy tree species, and the proportion of Fabaceae among the different families of canopy tree species goes up too. So what these two indicators suggest is that this region is characterized by much more equilibrium-based forests that probably have not experienced a long-term disturbance regime in the same ways that the fluvial dynamics and stuff are constantly creating disturbances in the upper Amazon. So much later than that, Hans had this idea and then brought me on board to mine this data set. At this time, when we published it in 2019, we were up to 1,946 plots in the network. And a lot of different taxa at various levels among all of the plots in the ATDN. We wanted to just go in and sample the database to see if any or how many of those plots would meet the basic qualification of monodominance, tropical monodominance, as defined by Cano and Lohman in the 1980s. That would be 60% or more of the individual trees in a stand are 60% or more dominant in terms of number of individuals above 10 centimeters DBH, and or 60% or more basal area dominance in the stand. And not to belabor this too much, but we had these 1,946 plots graphed out from a couple of different perspectives, but plot rank in dominance basically is good enough to look at. And there's your 20% cutoff. There's a 50%, with Hans wanted to use that. A 60% cutoff would be there, which would be the Cano and Lohman definition of monodominance. And basically, there were very few plots in this entire data set where there was more than 50% dominance by a single species. And in fact, above 60, all of these plots were from our Desimbe corymbosa plots in Guyana. So what about Guyana? Here's an aerial shot from the Upper Pataro Basin, which is in, like the western part of the country of Guyana is really the eastern extension of the Guyana Highlands. That sandstone tableau or table mountain country pokes into Guyana from its main center of intensity in adjacent Venezuela. There are several species in the genus Desimbe that occur in this area, and I'll say more about what it is in a moment. But some other actomycorrhizal trees that in certain locations can be quite dominant in the forest. This genus, Aldina, is a Guyana shield endemic that's actually in the Fabaceae subfamily Papilionoidea, which there's very few instances of known actomycorrhizal occurrence in that subgroup of the legumes, and Pachyraima dipterocarpacea, which is one of two dipterocarps that occur in the Western Hemisphere. This figure 2 is from a remote sensing study with ground truthing that we did with a grad student at Humboldt State, Rebecca DeGagny. We're at about a 20 by 20 kilometer pixel right in this area in the Upper Pataro Basin, where we're able to combine Landsat imagery analysis along with a lot of ground truthing of pixels on the ground to come up with about an 80% probability of predicting where these Desimbe dominated forest would be in this landscape, and those would be in green, and another sort of swamp forest dominant, the Micrandra, versus the non-dominated by any one species mixed rainforest. So there's a lot of Desimbe dominated forest in this region. That's the bottom line there. So about the main Desimbe species that we've been concentrating our work on. This is Desimbe corymbosa. It's in the Fabaceae subfamily Deuterioideae, which is important to recognize because this is a recently recognized subfamily out of the old Cisalpinioideae, in which pretty much everything that's in this subfamily is an ectomycorrhizal species, basically, of which all of them, with maybe a couple of exceptions in Asian tropics, all of them are Centro or Guineo-Congolian and West African genera. One, Desimbe, in the New World tropics. There are about 19 species of Desimbe known from basically the Guyana Highlands region. This one occurs in Guyana, and it forms really significant monodominant stands where regularly 50 to 70% of the stem dominance and 70 to 90% of the basal area dominance. And it also recruits by mass seeding on a huge level. So I'll say a bit about its clonal regenerative capacities, but it's also highly sexual too. Keeps its stand stocked with enormous numbers of seedlings and saplings, while the big parent trees every 5 or 6 years go off on the next huge mass seeding event, and it is ectomycorrhizal. So a lot of the work that we've done in bursts over the last 20 plus years is trying to get at life history traits of Desimbe corymbosa and why it's able to dominate like multiple hectare sized areas with this extreme level of monodominance with ample recruitment of conspecific seedlings right beside the parent trees. So our basic working hypothesis that we've been trying to pick away at, which I cannot belabor, has to do with it's essentially escaping Jansen-Connell mechanisms. So that means that a seedling or seeds from parent trees that disperse in the immediate proximity of the parent trees are not suffering high levels of mortality by host-specific predators and pathogens, and they're able to establish conspecific recruits right in the stand. If you want to see a place in the tropics that basically shoots Hubble's neutral theory completely in the bud, it's these kind of forests in Guyana. because when you see them, their composition over time is highly predictable. There's nothing stochastic about this at all. So anyhow, the trees are highly epicormically reiterative, both with roots and shoots. The large individuals become multi-stem, giant root mounds. They trap tons of litter in all these complicated interstices between all the shoots. They are innervated by adventitious roots that are covered with ectomycorrhizas, et cetera. So we think all these factors are coming to play to maintain this tree as a severe monodominant tree where it does occur. So if the ECM trees dominate, there's got to be ECM fungi, right? And actually, this goes back to my Smithsonian days, because when we were doing collecting trips into the mountains, we had to pass through the intermountain valleys to do these transects up the tepuis. And every time, when we're in the Pacrima Mountains, every time we're in a valley, we're in these forests dominated by these multi-stem trees. Now, I had a mycological background already, and the mushrooms that we would see when we'd walk through the Decyme stance were all ectomycorrhizal taxa, boletes, russulas, amanitas, et cetera. So that's actually what spurred me to go back and work on this for my PhD, that part. I'm leaving out a lot of the gaps for the sake of time. But just to reiterate, the ectomycorrhizal symbiosis is one of several types of mycorrhizae that occur in the world. These numbers probably need to be updated, but we know that there's multiple origins in plant lineages of this type of mycorrhizal symbiosis, and even more independent origins in fungi, mostly in the agaricomycetes. You know, the older view is that there was at least 18 independent origins of ectomycorrhizality, and some estimates by Teddersu and others say there might have been up to 60. What it's led to, the adoption of this type of mycorrhizal symbiosis, is evolutionary radiations in both the host plants and in the fungi. You know, circa 2008, there were roughly 7,000 fungi that were ectomycorrhizal, described across about 250 species, but you know, Greg Mueller also estimated at that time that macrofungal, ECM fungal diversity may exceed 20,000 out of approximately 50,000 macrofungal species that may be out there, of course, the majority of which are undiscovered and undescribed still, especially from the tropics. So a little bit more about the forest. Ectotrophic forest, that's the term Rolf Singer used a long time ago, and a lot of people don't like it, but I define it as closed canopy tall forests that are dominated by ectomycorrhizal trees. Well, they're all over the world, like most of the northern hemisphere. In temperate and boreal latitudes, of course they are. Pinaceae, Fagaceae, Betulaceae, and others are often dominant in north temperate and northerly high latitude environments. Southern hemisphere also, Fagaceae sensualata, with including the Nothofagaceae in temperate South America, et cetera. But in the tropics, it gets weirder. You know, we know there's a lot of ectomycorrhizal activity because of the dominance of Southeast Asian rainforest by the Dipterocarpaceae, another completely different woody plant angiosperm lineage that's strictly ectomycorrhizal. You get into Africa, there's some Dipterocarp representation more in the woodlands regions of South Central Africa, but a big swath of area in which members of the Fabaceae subfamily Deuterioideae are present and sometimes highly dominant at the species level in forests in Central and Western African tropical forests. Get over to the neotropics, of course, that Cordillera spine that runs down through Central America, pokes into northern South America and Colombia. We're talking higher elevation Quercus forests with trees and fungi that are derived from higher latitudes that have come down. And then, of course, what we've been able to document and a few others in the Guiana Shield region. Fabaceae, subfamily Deuterioideae, and two occurrences of the two neotropical dipterocarps, Pachyraimea and Pseudomenodes, which is over in southeastern Colombia. This is from a review article we did in 2018. Just to reiterate again, we went through all the literature we could find to where individual tree species were confirmed as ectomycorrhizal. Was there a study done where they extracted the roots and confirmed the anatomy of ectomycorrhizas on it, and or they sequenced root tips from a tree species that had ectomycorrhiza morphology and came up with sequences from ectomycorrhizal fungi? There's some differences between the four main tropical regions. Dipterocarpaceae, Fabaceae I'm pointing out here because, of course, of the 59 species of confirmed ECM plants, the majority of them are dipterocarps in the greater southeastern Asia. You move over to Africa where there's more species that have been confirmed in the plants, 120. More than half of those are Fabaceae, and they're all Fabaceae subfamily Deuterioideae. Get over the neotropics back down to lower numbers of confirmed so far. Now there are a bunch of known angiosperm and of course in some cases gymnosperm that do occur in parts of the tropics that are ectomycorrhizal, but are they major players in the environment? We don't know, but not really in the neotropics. But this is very much a work in progress. Just even knowing that there's 60 ectomycorrhizal plant species in the lowland neotropics right now is got to be an underestimate. How come, though, and I was taught this in my ecology classes and stuff throughout my earlier education, that when we were studying mycorrhizae types, yeah, we got tons of ectomycorrhizae in the temperate zone, but you get in the tropics, they disappear. You don't find the fungi, et cetera. Why was that? Well, part of it had to do with earlier studies where they went to the root systems of tropical trees and they analyzed them morphologically to see which kind of mycorrhiza type they had. Most of those studies came up with strictly arbuscular trees. But other folks in different parts of the tropics, sometimes going way back, even to the time of Pachillard, Rick in Brazil, late 1800s, early 1900s, were collecting and making specimens of fungi that were clearly in ectomycorrhizal fungal groups. And these things were in herbarium stuff, and nobody ever thought to challenge the old paradigm that ectomycorrhizae are lacking in the tropics by going to look at what fungal herbarium records might be able to tell you. So ectotrophic tropical monodominant forest has been largely overlooked, and these are the places where you have massive concentrations of the fungi associated with the trees because there's high host density, and many regions in the tropics remain unexplored. So one way that Kathy and I and some other colleagues tried to rectify this was early on in Guyana, we set up a plot system to do traditional repeated sampling of the sporocarps, the fruiting bodies of the mushrooms in the upper Pataro Basin. And we had really good, a lot of the green is all Decyme dominated. We had 3 pure dominated Decyme plots, and 3 we tucked into these mixed forest areas that were lacking completely in Decyme. And we repeatedly sampled these plots. We had hectare plots gridded off into 10 by 10 meter cells and went in there every rainy season for essentially 8 years in a row and repeated sampled for the fruiting bodies in the peak of the rainy season for like 6 to 8 weeks. So there's a lot that came out of this. We also did saprotrophic fungi too, which made it even more complicated. But I'm not saying anything about the saprotrophs right now. But after this study, and we published this in 2012, we had roughly 127 ectomycorrhizal fungi occurring in the Decyme plots across the Boletaceae, Amanitaceae, Russulaceae et al. And general collecting and so forth in those years and ensuing years, right up to 2023, in that same general area, we're up to like 174 species. So, and this slide here is from yet another review article we did, just to point out that certain lineages, like the Russulaceae and Ammonitaceae and Boletaceae and Thelepheraceae, are wildly overrepresented in any study that's been done in tropical systems that looked at ectomycorrhizal fungi. So we'll skip this other than to say this was our first below ground study we did, and after all that sporocarp data that we had and ITS sequences and stuff to compare it to, once we started going below ground and sequencing the root tips, 60% or so of the taxa that we got off the root tips from the very same stands were unknown to us. We'd never found them as sporocarps yet. So probably estimated fungal diversity just at the Guyana sites, 250 to 300 species probably. Just to show some cool, pretty pictures and get out of here. Of course, most of these are new to science, so it's a long slog getting them taxonomically worked up and described with multiple collaborators that are experts in particular groups. There's one, Inospe lilacina squamosa. A few of them are previously described endemics, such as Cantharellus guyanensis, which is still the only Cantharellus species described from like eastern tropical South America. It was described originally by Montaigne from French Guiana back in the 1800s. A number of them have wildly divergent morphologies from the genus that they're in. Small little Lactarius and Russula species that are pleurotoid. Pseudotelostoma, which is an Elaphomyces that has dispensed with animal mycophagy and is sending up the spore mass on a stalk to be battered by raindrops. Resupinate clavulinas, tiny little cantharelloids, which are actually Craterella species, this one in particular, that look very much like discomycetes, that are actually tiny chanterelles with a smooth hymenophore, and the world's tallest Cortinarius altissimus, which gets up to 30 centimeters long stipe. New genera, we've described a number mostly in the Boletaceae, and we've found some new records in certain groups, mainly the Elaphomycetaceae, that were literally the first records for Elaphomyces. Really common mycophagy dispersed Ascomycete group in temperate forest, poorly known in the tropics, unknown from the South American continent, not just the South American tropics. We got 3 species of Elaphomyces there, and we're still working on this stuff. We're right now describing Cortinariaceae. That paper just came out in Mycologia, and working on the Thalepharoids too, and getting some of those described. So that's it. Did I make it in time?
Okay, I think we should probably go ahead to the next speaker.
Good.
All right. Very excited to hear your talk, Kathy Aim, and have heard so much about you. So this will be great to have another speaker who's acquainted with the Central Guyana Shield, but we actually asked her to talk about rust fungi, so we would have some diversity. And Kathy Aim is going to talk about innovative use of collections, a few case studies in the Punchinales, the rust fungi. Oh, sorry. Oh gosh, I'm going to have to make this up. Okay, there you are. Okay, so Kathy is professor of mycology at Purdue University and director of the Purdue University Herbaria. And so she's director of the Arthur Fungarium and Kriebel Herbarium at Purdue. She received her master's and PhD in biology at Virginia Polytechnic Institute and State University, and did postdoc research at the University of Oxford. And her work combines expeditionary field work and traditional approaches with molecular genetics and multi-omics approaches to understand fungal diversity and evolution. Thank you, Kathy.
Well, thank you very much. Janine, Don, Jeanette, everybody for making this possible. I love to come to the Farlow. It's so rich, and I love Cambridge as well. So I haven't been here post-pandemic, so it's really nice to be back and to see some people again. And just seeing Terry's talk got me really excited to go back in the bush, which every year when we leave, we say we're never going back because it's so hard to get out there, and then I just die to get back. But what I was going to talk about today is something a little bit different. Because we are talking about the Farlow, and I do direct some herbaria, I've recently done a stint at the National Science Foundation, and one of the things we're trying to push at NSF is additional funding for collections and for research that utilizes collections. A lot of the data, for instance, that we've seen earlier in other talks, David showed, for instance, data on mycoportal. And those are all giant data aggregation collectives that were funded by the National Science Foundation to get these data onto public repositories where we can use them and find new ways to use collections. So what I'm actually, it's a small part of what we do, but we're used to using collections for obviously floristic works, taxonomic works, monographs, for looking at biogeography and all these secondary and tertiary associations of these collections. But what I want to show today is some sort of non-traditional uses that we can also use, or other ways to gather data from collections to maybe solve some other long problems. And this is actually a new DCL for any of you who are interested in doing collections, unusual sorts of collections-based research. At NSF, we now have a DCL out for thinking up innovative ways to use collections for this kind of work. So a little bit about the Purdue University herbaria. What drew me to Purdue was actually this collection here. So this is the Arthur Fungarium, and the Arthur Fungarium has what is likely, we like to consider it the most important collection of rust fungi in the world. Rust fungi are this order called Puccineales. And I'll talk a little bit more about rust fungi in a minute, but this is one of the areas I specialize in. And rusts are incredibly important. They cause some of the most economically important diseases. They are changing landscapes. Emergent rusts, for instance, in New Zealand and Australia are completely changing the ecological landscapes. They are wiping out plants, etc. Natural landscapes, as well as in Hawaii. So they're extremely important, and there are about 8,000 described species of rust, which make it our second largest known order of fungi in terms of described species. And yet, this is something I've heard a couple of times talking out in the hallway and in here, like when Janine talked about this big gap, 2.5 conservatively estimated fungi. How are we going to close this gap? Something like rust, the second largest order of fungi. I am the only non-retired person in the Americas who studies rust. And there are two other people who study rust systematics, and one will be retiring next year in the world. So we're just about down to two people to study this huge group. And you can repeat this story over and over and over in fungi. We're just lacking a lot of individuals to do this kind of work. Now, in addition to the Arthur, which contains some 5,000 type specimens of these rusts, and you all remember Don told us about types, but this is the specimen that anchors that name, the application of that name through time, so that we always have something to compare names to. So types are extremely important, especially in something like rust biology, where we're talking about very small microfungi. The Kriebel collection is also important as the oldest collection in Indiana. It was founded by one of the original 6 faculty members of Purdue University when Purdue was established. And Kriebel right now is especially rich in tropical fungi because, of course, I've been adding to it, but also in other plant pathogenic fungi groups. So we have very rich collections of other plant pathogens. We also, very interesting, so this mycoportal initiative that came out of an NSF grant, we found by cataloging and databasing, and you can't, I see over here, I just want to point out Cade Kane over here, who is an undergraduate in my lab. Glad to see you, Cade. But Cade did a lot of this databasing, you know, all those data that are on mycoportal. Some poor undergraduate had to go in and database all that stuff. Yes, thank you, Cade. But in the process of databasing this stuff, we discovered that we actually have the oldest fungal collection that we know of in North America is at the Arthur. And it is a rust that was collected on Captain Cook's first voyage. It was collected in Tierra del Fuego. It's on a Berberis, and it is, again, a rust specimen that was sent to the Arthur to be identified because we have this long, long history of people who work on rust fungi. So, of course, as I've mentioned, we use these collections all the time for everything as mundane as making identifications for plant diagnosticians or people who are facing new emergent diseases around the world and need positive IDs. We use them for floristic studies with rust because these are obligate pathogens on plants. We can look at the collections to get host range data. We can, of course, sequence these now. These are uses, of course, that nobody envisioned when these collections were made, but sequencing them, looking for cryptic species, all these traditional uses. One of the things I want to talk about next, though, is some of the non-traditional uses, and this is solving disease epidemiological questions. And what you're looking at on the far right of the screen is coffee rust. You all know about coffee rust and how important this is? So coffee, of course, is our second most traded commodity in the world after oil. It's extremely important. And coffee is grown around the world primarily by small shareholder farmers in tropical regions of the world. We believe that the crop originated, or the plant originated in Ethiopia. At one time, the British, during their colonial expansion, had converted most of Sri Lanka to growing coffee, and were huge, huge exporters of coffee. And then something happened around 1870, and the colonies in Sri Lanka got this disease, which we now identify as coffee leaf rust here on the right, and it completely wiped out the crops, and people started moving the crop around the world to outrun this disease. And a couple of years ago, I would have told you that Hawaii was the last place on the planet that didn't have coffee leaf rust, but it did manage to get to Hawaii a couple of years ago. And trying to understand a couple of things about this pathogen, it's really stymied traditional uridinologists. So what you're looking at here, for everybody who's not familiar with it, rust fungi have what we call a complex life cycle. So these types of life cycles have actually arisen a couple of times in the tree of life, mostly in animal pathogens. To my knowledge, rust fungi are the only plant pathogens that have adopted this, what we call complex life cycle. And what that means is these things undergo separated gametophylae and sporophylli. So they do an alternation of generations. They produce 5 different spore stages during this life cycle. And each one of these spore stages, the spores are cytologically unique. They have a very different function, very different morphologies, of course, different ways of dispersing. And what most of our rusts do, in addition to forming these separate gametothalli and sporothalli, is that they'll form these different thalli on different hosts, and these hosts are not related to each other. And every species of these 8,000 or so species of rusts that we know about, those that do the host alternation, those plants are ecologically co-distributed, but they have nothing to do with each other, of course, phylogenetically. With coffee leaf rust, this is one of those species, because of this complex life cycle. I forgot to mention that you can't pure culture these because they are obligate pathogens. And this makes doing molecular work very difficult, as well as doing any kind of controlled experimental work when you can't pure culture them. And the genomes for these things are monstrous for a fungus. Maybe not for a plant, but they can certainly be up to a gig in size. So these are really, really big genomes with lots of repetitive DNA. So again, very, very difficult to piece these things together. But anyway, because of all these problems or complexities to the life cycle, there are many, many rust species for which we don't know the entire life cycle. And the fact that coffee leaf rust is the most important disease of coffee, it causes the most damage and limitations to production in coffee, we still don't know the entire life cycle. So this is one problem that we have. So we know that on the coffee leaves, it produces these special sori that we call uridea or uridinea, with uridinospores, and these spores can blow back to the coffee plant. They require about 100% humidity to germinate, and they germinate through the stomata, which in the coffee is on the underside of the leaf. So there's some environmental conditions that have to be met in order to cause an epidemic, but when it gets going, they can continue to cycle in this uridinospore stage indefinitely until those environmental conditions are no longer met. What usually happens with rust fungi is at some point during the growing season, the environmental conditions change. Usually they get drier, and this causes the cessation of the production of uridinea and the production of a new type of soricin spore that we call a teleum with the teleospore. And these teleospores in temperate regions, they will overwinter. So they act as a spore for overwintering and then germinate in the spring. In a tropical condition, they'll just wait until conditions are wet and moist and humid again. And then these teleospores themselves actually germinate into what we call basidium. David talked about basidia earlier, these ballistosporic things, but it's inside the basidium where meiosis actually occurs. Both karyogamy and meiosis occur, so it's a little different than what plants and animals do, one following the other. But the result is you get these haploid basidiospores, and then those go off to the alternate host. You get two haploids that are compatible. They will mate on the alternate host, form another spore that's called an isospore, and that goes back onto the coffee plant. But again, we have no idea what that alternate host is. And so this raises a lot of questions, especially for breeding programs and trying to manage the disease. Because if you are looking at something that is just purely producing clonal spores, that's a different management strategy than something that's undergoing sexual recombination at the end of every growing season. This would require some kind of durable resistance that would be built in to overcome any recombination of newly evolved resistance genes. So this is a big problem, again, with coffee rust, not knowing what this alternate host is or where it can be found. And there are a couple of ways that we can go about trying to do this and have been trying to figure this out. I went to Kew Gardens once and looked at gazillions of herbarium specimens to see if I could find a spermagonial or eschal stage on a potential alternate host. We've got coffee growing in the greenhouses now, and we are trying to inoculate that onto potential alternate hosts. And I could spend another lecture talking about how we're trying to pinpoint down what the potential alternate hosts are. But another way, of course, is to do this indirectly and infer it from molecular data, perhaps, and at least see if there's any evidence there that we have what looks like sexual recombination. That would tell us that there is an alternate host and where it might be if we can pinpoint those locations. These are just some fun graphs from old USDA pamphlets, but showing in 1952, we believed, of course, that we were safe over here on our side of the world from coffee rust, that the Atlantic would form a big barrier to dispersal. And of course, that didn't happen. And as I said, it's gone all the way out to Hawaii as well. And another question we have is how is it moving around in these cases? And we can also use population genetics to give us a hint at that. So what we ended up doing first off was just getting a lot of modern collections. Because these genomes are difficult to obtain, we started out with SSR work. We couldn't get a good genome way back in the day when we began this work. And we had only about 12 SSRs in this initial study that were really working, because one of the things that was amazing is there wasn't a lot of genetic diversity in all these isolates. So here are the places that we got the isolates from, and here's just simple network analysis, but you can look at this and see that it looks very clonal, right? There's two, this one's mostly Eastern Hemisphere, and this one's mostly Western Hemisphere. There's two sort of aggressive genotypes that are found in most places, depending on the hemisphere, but very, very clonal. And we did lots of tests to show that there was no evidence for sexual recombination occurring, at least within the areas that we were able to sample. But of course, we were missing a lot of detail. One is the place, here's Ethiopia, where coffee is thought to originate, Café Arabica. And we had no idea what the genotypes look like around here, around this point of origin, at least for coffee. And one hypothesis is that Hemilia vastatrix, coffee rust fungus, also originated there. So we needed to test that area. We needed to test Sri Lanka, of course, which is where the disease was first made its appearance. And Brazil, which is where it first landed, so we think, in our hemisphere, in Brazil in 1972. And so this is where we were able to utilize herbarium specimens to give some sort of time scope to this, as well as utilize material from places in the world where we couldn't get. So here's our newest sampling regime. And what's really, really cool about it, look at all this diversity here. These are pretty much all single genotypes. All of a sudden, we see all these single genotypes, and these are all, or a single sample, I should say, genotypes from a single sample. They're all coming from this area right around Lake Victoria. So we have Rwanda, Uganda, and Kenya, where we have a tremendous amount of diversity there. Again, just single MLGs, not clonal representatives. Ethiopia, surprisingly, is not showing quite the same pattern. So we think that the pathogen actually originated around Lake Victoria and then was spread from there. We see one, actually, I'll just make this a lot easier on us. There we go. So what we're seeing is from this area, a spread over to Sri Lanka. We have some historical Sri Lanka genotypes now, and these match very much the genotype. Is that working? Aha. I can make this work. Okay. So our Eastern Hemisphere genotype over here has moved somewhere from around Lake Victoria into Sri Lanka, on through Asia. And additionally, this is where our Brazilian introduction is coming from as well. The Brazilian introduction undergoes some mutations, and pretty much moves clonally all the way up through South and Central America. And what we see, this is again from another old USDA handout, but that our genotype data is matching very well the known route of dispersal of the fungus in the Eastern Hemisphere at the time. Where it doesn't match perfectly is dispersal over here, because what we're seeing is signs of actually 2 introductions in Puerto Rico and Brazil instead of just 1. We are seeing an introduction actually from this part of Africa as well, so more like Western Africa, into Puerto Rico and into Brazil, but those don't seem to be very aggressive genotypes, so it's mainly the main genotype that has been moving. And we have also, it's not on this map, but Hawaii also originated from Central America. It's the same genotype as Central America, not from one of the Asian genotypes that we see. And it turns out that in Hawaii, they have a practice of hiring migrant workers from Central America during coffee picking time. And these spores are probably adhering to clothing when they move. Okay, I'm already almost running out of time. So the other thing I wanted to talk about is a study that one of my graduate students, Paula Gomez Zapata, did. And Paula was really interested. So as I mentioned, rusts are extremely important economically, agriculturally, environmentally, but we're really lagging behind in control measures for them other than copper fungicides and things like that. And Paula was interested in trying to screen these things for hyperparasites, things that could be used as potential biocontrol. And so there's one of our herbaria cabinet with about 200,000 rust specimens. And like David showed, these specimens are kept in packets, and we usually have up to 12 packets on a sheet. And you have to actually physically open these up, and here's the specimen inside. And this is a particularly attractive rust because you can really see the sori on this thing. But it's a bit laborious to get these things open and start screening them. But what Paula did is she screened about 8,000 specimens randomly selected and looked for the presence of hyperparasites. So these are parasites of the rust fungus that were accidentally co-collected along with the rust fungus. And she found all sorts of things. This is Spherulopsis, so this is your rust sorus here, and this is black stuff is the hyperparasite. This is a Lecanicilium, which is the white stuff. Here you can see an uninfected rust sorus and a little bit of this one that's infected here. This is a Tuberculina. This one is an insect larva. That's a fly insect called Mycodiplosis, and these larvae feed on rust spores. And then there's a Ramularia, and this one I'm going to talk about right now, Trotula. But at any rate, yeah, here are her data. So she screened 7,000 specimens in 99 rust genera, and a little more than 10% had these hyperparasites. Now, the same thing when you make plant collections, you tend to try to pick non-diseased leaves. And the same thing when we make the rust collections. If they look superficially moldy, we tend to throw them. So this is obviously going to be probably an underestimate of the amount of diversity of these things that occur. But it is a good way to use herbarium collections just to get an idea of what's out there, because most of these individual organisms we really didn't know much about. Paula did, of course, the microscopy, and then we did barcoding and phylogenetic loci as well. But one of her interesting discoveries, and I bring this one up, it's the genus Trotula. And this is a little Ascomycete genus that's thought to be saprotrophic on plants. But here is the rust, and here's the Trotula fruiting on it. And you can see in all these cases, it is fruiting just on the rust sorus here, these little fruits. There's only a couple of species of these things described, and it turns out that this was a new species, so it's a new trophic habit that we know of, the first time we know of this genus being mycoparasitic. But what's kind of cool is the specimen. It was collected in 1912 from Trinidad, and it was part of Roland Thaxter's Farlow hexacati that she found this on. Yeah. So there you go. He had no idea he was collecting a new record of a hyperparasite at the time. This is the genus Sphaerolopsis, and we don't know much at all about this genus. There's 5 or 6 species described. And again, we know that some are saprotrophs, but some seem to be associated, again, with rust sori. So here's a sorus, and here's the Sphaerolopsis. But what Paulo was able to do, and here's a beautiful, this is actually a rust spore. So this is a Puccinia. So you can see the Puccinia sorus here and the Sphaerolopsis fruiting right in the middle of it. So what she was able to do is answer a lot of questions about the distribution of this genus just by doing this survey. She was able to discover a new species. It's quite broadly distributed. But she didn't find any evidence of host specificity, which is one thing we didn't know at the time, but these don't seem to be specific to a plant host or to any particular genus of rust, and no real strong signal of geological specialization either. But one thing, so we did show that this genus is very, very widely distributed around the globe. So we have much more information on its distribution. And if you can see this, showed that it does appear to be antagonistic. This is actually complemented with an in vitro study with rust spores, corn rust that we grew in the lab, and then she inoculated with Sphaerolopsis. But these blue coils are actually a Sphaerolopsis coiling around the germ tubes of a rust spore. So they're definitely antagonistic, and through time actually sucking all the contents out of those rust germ tubes. So it's very cool the way she was able to couple greenhouse studies with her studies. And then the last one, Mycodiplosis. These are the fly larvae that feed only on rust or powdery mildews. And so this is what they look like when they're accidentally dried with a rust specimen. Not very attractive, but here's one actually feeding on rust spores. They're really, really cute. There's about 40 species described, but they are impossible to identify without the adult male fly. The adult male is microscopic, and it's really difficult. You can bag the leaves with the larvae. The larvae drop off and pupate, and then hope that you get some to come out, to emerge, that you can then identify. But anyway, because I'm running out of time, here's Paula's work on Mycodiplosis. We were able to match names to some of these by getting emergent adult males. But basically what it shows again is there is no host specificity with these, but there may be some geological specialization. We've done the same thing with insect collections. So Don mentioned Labubaniales, but we have an insect collection at Purdue as well. And this is Paulina, who has been looking for labules on rove beetles, and so far found one new species of these little ectotrophic fungi. Okay, I think I just made it in time, so let me thank my lab. Sorry for running through the end of that. Thanks for your patience.
Kathy, I think you can take one question.
Okay.
One question, guys.
You give one sentence and you give one sentence.
I just wanted to know, that's such a complicated life cycle. I just want to know if there are cases known of intermediate versions. One of those things like they always talk about, it's so complex, how does it get there?
No, that's something we're trying to do. Yes, so there are known life cycles that don't follow that. But whether this was a step-by-step or a reduction, we don't know yet. The key to that is actually the oldest extant rust we know is on Torreya californica in California. It's an endemic conifer. And we only know one stage of the life cycle from that. So the ancestors had Telea spores. Anyway, long story short, it's the Uridinea spores I can't place until I can find the alternate host for that one. And then we can tell you whether it was incremental or one giant boom. Okay, thank you very much.
Thank you, Kathy. Our next speaker is Rebecca Yar, and she's come all the way from the Royal Botanic Garden in Edinburgh. And she's going to be talking today on lichen diversity conservation baselines from collections. Rebecca is the lichen biodiversity scientist at the Royal Botanic Garden in Edinburgh. Her research focuses on the diversity, detection, and conservation of lichenized fungi using traditional survey, taxonomic, and genetic tools. She's the chair of the IUCN Species Survival Commission Lichen Specialist Group and associate editor of the Lichenologist and Edinburgh Journal of Botany. Thanks, Rebecca.
Well, it's really exciting to be here. I'm really proud to be part of the Farlow's 100th centenary. That is just pretty great. I don't want to do one-upsmanship, but we just had our 350th in Edinburgh. It's been around a little while. It's also really a pleasure to be in an audience, have an audience full of people who know about mycology, so that's great. So today, as Janine already mentioned, I'm going to be telling you stories about lichen diversity. I'm interested in conservation, and we're going to be talking about using lichen collections, preserved historical collections, to tell you about developing conservation baselines. And that's all about measuring diversity and comparing that across time points. So the talk is in a few parts. I'm going to do a really brief introduction about lichens, and then I've got 3 case studies about trends in time from the past, from the present, and the future using 3 different systems. So you probably are all familiar with lichens. They're fungi that form perennial structures. They're often from the cup fungi, like this. This is a non-lichen fungus here. And they produce these wonderful little microscopic spores with a change in humidity. I don't know if anyone's done that experiment. You go, and then you can get the puff of spores. It's really fun. But most lichens are much smaller than this wonderful cup fungus. So these little fruit bodies are only a couple of millimeters. So they'll often produce spores inside those fruit bodies, but they can also reproduce asexually, forming peg-like structures or powder or grains. Lichens are found, of course, on all the continents. They occur from the tops of the mountains to below sea level. They are recognized as being pretty tough in some ways. So if we think about a single lichen, why am I here in this sort of fungi fest? So here's a lichen growing on the coastal rocks in Scotland. And if you do a longitudinal section, what you can see is the outer layers here on the left and on the right are these consolidated only fungal tissues. They're protective layers, but they're also metabolically active in absorption. Just inside that, you have the photosynthetic layer. So these are single-celled green algae in this case, and that's, of course, the root to the carbon source. So these are photosynthetic cells that are taking carbon dioxide, gaseous carbon dioxide from the atmosphere and releasing gaseous oxygen using solar power, as all photosynthesis works. And the lichen has a special structure, so these really loosely woven fungal threads in the center where the gas exchange happens. So these thalli, as we call them, are quite structurally complex to allow their photobionts, their photosynthetic cells, to be displayed to the sun and promote that gaseous exchange. So we know they're symbiotic. We know they're photosynthetic. We know they're microscopic. They have microscopic thread-like cells. These are so small they can penetrate within the grains of rock, so the rock looks like it's just been painted. You can't collect a lichen like that unless you collect the rock with it. But lichens are also osmotrophic, as most fungi, all fungi are, as far as I know, which is that they absorb their mineral nutrients and moisture from the outside surfaces, and that allows them to be pretty delicate in some ways. So even though we think of them as being tough, being on the tops of mountains and under seawater, they're also really delicate because they can't help but absorb whatever hits them, basically. The diversity of lichens worldwide is about 25, 30,000, something like that. And in some places like the UK, where I'm working, they're really well studied. And these combination of traits allows us to use lichens to sort of understand their habitats. So they're tough, but they're also really delicate in some ways. They're very specific for their tiny niches where they grow. This is a rainforest lichen, and it has a cyanobacterial partner, not a green algal partner, very sensitive to pollution, and needs a ton of liquid water. So they're all different. There's a lot of reasons to study lichens. I've already mentioned sort of using them to understand their environments on a micro scale, so they're great bioindicators. They also have interesting patterns of global diversity. They have local importance in nutrient cycling. So there's a lot of carbon being fixed here for little critters to eat, but also some species, like I just showed you, are nitrogen fixers. They provide ecosystem services, including the nitrogen and carbon cycling, but also other things like mediating water flow. And they're great model systems. So there's a lot of genomic attention paid to them right now to understand how the symbiosis actually works. So at the Botanic Garden, I mentioned it's been around a little while. The lichen team started with Brian Coppins in the 1970s, at least most recently. We've got historic collections as well. And I work really closely with him. He's one of the preeminent lichen taxonomists and ascomycete taxonomists in Europe. He retired a few years ago, but still very active. Chris Ellis is one of our, he's an ecologist, an ecophysiologist. He does lots of statistical modeling, and that's really useful. And we have a whole series of people coming and going, postdocs and PhD students and technicians, and different kinds of funding streams. So what I want to do today then is give you 3 different stories about these conservation baselines. The first one is from the past, and it's thinking about biodiversity loss through the transition to industrialization. So cast your mind back. This is what medieval England looked like. This is what we all did back there in medieval England. We hit trees with sticks. And the idea of this is, of course, to knock the acorns off the trees and feed the pigs. But really, I want to show you this to just think about what pre-industrial land use was like. So population of humans was low. The impacts of people on the landscapes was generally low. There were not a lot of people in the cities. And so it was a very different environment than what we face now. So let's fast forward to 1952 in London. And this was the Great Smog of London. That was a single event where pollution combined with fog created these terrible atmospheric conditions. And that single day was estimated to have killed 10,000 people. So this, unfortunately, still exists in parts of the world today, especially in Southeast Asia. Air pollution has been mediated and ameliorated in most of Western Europe, and we don't get this anymore. But if you just think about the changes from here to here, you can imagine the effects on biodiversity as well. So let's think about a timeline. So here's the history of medieval England moving forward to today, somewhere down here. And I've blocked off the industrial period. In Western Europe, in the big cities, this started in about 1750. Now, unfortunately, that's also when the discipline of biological science as a taxonomic framework with the classification system was developed by Carl Linnaeus. So this is the publication of Systemae Naturae. And this was the first time that somebody had sort of systematized the diversity of life. At that same time, it also gave rise to a whole new discipline of science, which is taxonomy. So this is the number of taxonomists through time. So here's Systemae Naturae, and this is today. We've got a lot more people working on this as a discipline. Because of that proliferation of interest and having a system to work with, we've unfortunately been able to detect a terrible decline in biological diversity overall. This is a figure from the World Wildlife Fund's Living Planet Index. And it tracks changes in global populations of about 5,000 species, mostly with backbones, from 1970 through, this was 2018, and that's a 69% decline. So this is the decline of diversity over time. Recognizing that problem, a bunch of people, a bunch of biologists especially, but policymakers too, got together and in 1992 devised the Convention on Biological Diversity, which was signed by 192 different countries around the world. That gives a legal framework and a mandate for protecting biodiversity. Great. So that starts here. We have the science starting here, but we have a bit of a question, don't we, about what was happening before those effects of industrialization. Now we know the effects of industrialization are massive. Geologists have named an entire new geological era called the Anthropocene, reflecting human population increases, extractive materials resource use, CO2 emissions, and of course the decline in wildlife. So the Anthropocene has had a massive effect. And if we kind of cast our minds back and try to think, how can we reconstruct what we've lost? Well, unbelievably, lichens could be an answer. So we have lichens as an archaeological resource. That's based on 2 observations. You probably have all seen out there in the Cambridge, is it called the Cambridge Common? The wonderful common out here, the trees are covered in lichens, right? Trees support lichens. They grow on the bark of trees. Trees are also used to make old houses. And across lowland England, you have houses like this, built from the things that you could see from standing on the roofline. So this project was conceived by Chris and Brian to try to understand the amount of diversity that was preserved in old buildings. An old building like this was built by using big timbers to make the framing, and then these panels on the inside are woven sticks with mud slapped on the outside. So this is wattle and daub on a timber frame building. In a single county in England, there's about 4,000 of these thatched cottages remaining. So the resource is tremendous, but it had never been explored. If you think about the actual process of conversion of a timber tree like this, so they're grown in woodlands like this where you have standard trees, the big trees, and then coppiced woodland around. So these are hazel stools, which are perennial woody structures that give rise to new tree sprouts, and they can be cut over and over. And this kind of traditional management has been done for thousands of years probably in Europe for the production of fodder, but also as building materials. The big timbers were processed by hand. You don't want to be the guy at the bottom of the saw, right? In the pit, that would be a terrible job to have. It was hard work, and they were roughly squared only by sort of hand axes if they needed to do that, because there's no reason to have a perfectly squared timber if you're going to build a house. A small, low-status house doesn't need perfectly squared timbers, and it opens the possibility of having these curved edges with preserved bark. The archaeologists that we talked to said, oh, you're not going to find any bark. They used it for tanning. You know, it just won't be there anymore. Good luck with that. So we persevered and looked at about 100 buildings, had cups of tea with owners in places like this and beers in places like that, and then climbed up into the roof spaces. And that was not exactly a pleasant experience. It was very dusty up there. But people were super excited. These guys said, come back on Thursday. And they had torn a hole in their roof so that we could go upstairs. It was quite amazing. But as a result of this, we've ended up with a whole bunch of new preserved collections, actually very old new preserved collections. So we had permission to take these samples back, and they're now incorporated as part of the Royal Botanic Garden Edinburgh Herbarium, E. And these are now our oldest collections. These are from the 15th century. So these little bits of roundwood have their bark on them, and we can still find lichens preserved. So here's what they look like. So sometimes they still look like lichens. Sometimes they just look like dots, but that's what a lot of lichens look like. In Britain, we have about 75% of our species are like dots. We looked at about 100 different buildings across lowland England. And this is where Brian Coppin's expertise is really important. So he developed a huge amount of our modern collecting in the herbarium at the Botanic Garden. His collection numbers are in the 25,000s right now, so he's done a lot of collecting himself. He's really familiar with the diversity and helped a lot when I got stuck. So here's what something looks like now, and that's the historic material. They don't look exactly alike, but what's interesting is that the spores and the chemical reactions were preserved really well. So the ability to identify things was actually not particularly badly impacted, and we had managed to identify nearly everything. Here's an example. This is a little orange-fruited Caloplaca species. It was found here in not too far from London. But this is a map of the current distribution of that species generated from British Lichen Society data, so shout out to all those people who submit collections to your local Natural History Society. We have about 2 million records on the British Lichen Society database now, and it allows us to look at those distributions really well. And if you just look at this, you can see that species is definitely much more northern and western than where the historical collection is. So this is a plot of the 95% confidence interval of mean annual temperature of the current distribution of that lichen, which is sitting around 7.3 degrees Celsius. And that's the historic site, sitting at 9 degrees. 2 degrees C makes a massive difference for mean annual temperature for species ranges. So this is a clear signature of a climate change difference. And that corresponds well with what we know, with what's going on in the UK and Western Europe. We had the Little Ice Age in the 1700s, so that fits. Here's another example. This didn't look like much at all until we looked at the spores, and the spores are massive. These are 80 microns to 100 microns with beautiful patterning, so that's all preserved really nicely. The current distribution looks like this. It's all over the UK and Ireland, and the spot that's down here was where the historic collections are from. And you can see here there's a gap in the middle of England up to the industrial parts of the north, and that corresponds really closely with high pollution areas. This is where the most industrialized parts of Britain are. So this is a signature of pollution, and therefore this is the 95% confidence interval for sulfur dioxide concentrations. That's the main contributor to acid rain, to which lichens and bryophytes are very, very sensitive. So you can see these guys are well outside the comfort zone of sulfur dioxide. So we could just do that for individual species, but we wanted to look at the whole data set taken together. And if you think about what we can infer as loss over time, if we've got something that we found in the historic record here, present in the historic record, and present in the modern day, that's no change. If we never found it in the historic material, there's absolutely nothing we can say about that. But if we found it in the historic material and we did not find it in the present day, then we can infer a loss. So that's sort of the most interesting part of the data set. But inferring that loss requires some confidence, right? So we're going back to the British Lichen Society data here. So if our target area is here, just because there hasn't been a dot there in the last 60 years doesn't mean it's not there. So we need a statistically confident kind of inference of what's lost and what's not lost, or what was present and not present, if you see what I mean. So we developed a threshold for for present day presence and absences based on sort of a GIS analysis. And using that, we came up with these incredible numbers. So for 3 target villages, I'm standing in people's way, I think, up here. You've got your other. So in 3 different target areas, we have losses between 80% and 35% of biodiversity in these areas over that industrial threshold. That's huge losses. Imagine going to your local woodland and 80% of your birds are gone at the species level. That's a major change. So we have a quantification of biodiversity loss. And I'm going to add a little nuance to your understanding of this by telling you about our prevailing winds, right? So this is the west coast of the UK, and so this is the Atlantic Ocean. The Gulf Stream's coming out this way and bringing warm, wet air over here, and the southwesterlies are pushing the pollution from London straight into this spot. So that 80% loss really corresponds nicely to what we expect from pollution patterns in the UK. So how else can we explain these losses? So we know already that pollution is important. So this is the overall pattern of sulfur dioxide deposition across the UK, and that 95% confidence interval sits here. And the 3 villages we've sampled are well above that. So pollution is a major effect for all of these places. It was across all of lowland England. But if we think about other moderating factors, this area here in Wiltshire is known as an area with lots and lots of forest. That's not a green-looking sheep. That's actually my cartoon of forests. So here in Wiltshire, we have a lot of forests, and we think that even though the pollution situation is fairly similar in these 2 sites, the amount of the extent of that ancient forest probably filters the pollution to enough of an effect that it affects the amount of loss, and it mediates that to some extent. So let's go back to this overall loss of 70% using the Living Planet Index from the World Wildlife Fund. So that was that declining dark green line I showed you before. This is now taken apart by geographic area by continent. So in Western Europe, that decline is about 20%. In North America, it's about 20%. So keep in mind that the baseline here is 1970. So pushing back that baseline further would make a really different picture. The losses are not as steep here because, of course, the losses happened a long time before 1970, but the data weren't good enough then. So the big declines are now happening in the global south. And it really highlights that problem of having a conservation baseline from the wrong period. We don't want a conservation baseline from 1970. That would be the worst time for the Western Europe because that's when the peak of the sulfur dioxide was. And so there was almost no lichens in most of the cities and throughout a large part of most of urbanized Europe. Okay, so let's move forward. We're going to now think about using collections for biodiversity baselines for present day studies. And I'm going to give you a little geography here, just so you know how big the UK is relative to wonderful Massachusetts. So that little line there, that's Scotland up in the north. This is 8%, by the way, of the land area. So here we have Scotland. The big cities are here, Glasgow and Edinburgh. This is where most of our population sits. This is, of course, the winter time, and you can see all the snow sitting and picking out the mountains. It's a really mountainous area. We have not a lot of people and difficult access, quite frankly, even though it's not a very big area. If you've ever tried to take a family holiday to drive to north here, it will take you a lot longer than you think. So don't hire a car and expect to get there at the north in a 2-hour period. You'll take at least 6 hours to get there. And if we think about one of our highest massifs, so this is the Cairngorm Plateau here, and those are some of our highest mountains. They get to 4,000 feet-ish. Here's some of the mountains: Cairngorm, Ben Main, Ben Macdui, Ben Averd. This is a 17-mile walk from the closest village. It's not Guyana, right? I know it's not as hard as that, but it's not an easy place to go and survey. And these mountains, although they're not particularly tall from the point of view of tall mountains, it's Arctic tundra on the tops of them. And even in the summertime, you get snow and you get ridiculous wind, and it's a really difficult place to work. So this is a picture from late spring, and all this kind of creamy area should be white. That's all snow. So these are patches of late-lying snow. They've not been surveyed particularly well. They're not easy to get to, and the weather is horrendous. So I'll just give you a sense of the scale of the challenge. This is a little tiny chip of rock that I collected up there. This is a coin about the size of a nickel. This is a large area to try to pick out every nickel-sized piece of rock and look at it. But that one was collected because of this. So there's a really distinctive looking thing. I'd never seen it out there, and lots of wanderings. Picked it up, was excited about it, and then found this, which was thought to be extinct for the past 40 years. So these species, now you can see all these little difficult to see words and spots. So those species are not all that uncommon. It's just that it's pretty hard to get up there. And there's not a lot of people who can go up there and identify these things. Poorly surveyed, not very well known. You would think that in the UK, it's one of the best known sort of floras, loosely speaking, fungus in the world, but we've got lots of things that are just still poorly known, which is where barcoding comes in. Some people have been mentioning barcoding today. I'm going to walk you through it really quickly. Just like you can scan your can of soup and at the grocery store, those little bunch of lines tells the machine what you're looking at. Little sequences of DNA hopefully tell you what you're looking at. And in about 70 to 80% of fungi, a short diagnostic sequence will correspond to species level identifications. So you can start with your little chip of rock here, extract its DNA, generate a DNA sequence, and line up all your A's and T's and G's and C's so they look like stripes, like that. So there's your barcode. You can submit that to an internationally freely accessible database with the goal of comparing your sequence with other species sequences that are in that database. Is it species D, or is it maybe a new one? And this has been a successful venture over the past few decades. We've got about 19 million barcode sequences in the Barcode of Life database, for example. And then you can use your short sequence to try to identify things. Now, why is this important? I think one of the main things that barcoding is important for is connecting expert users and a well-identified little specimen like this with lots of other people who are going to want to understand biodiversity distributions and data, but can't do the identifications. I would definitely support the idea that very many more people can generate a DNA sequence than can identify a lot of different forms of life. So we undertook a little barcoding work up in the areas where the snow lies late. It was only about 300 specimens. We did all this barcoding stuff, and we found so much new stuff. It was really surprising for this area. So this is about 1 square centimeter of rock, and all that little pillowy white stuff is a species only described from North America, and so far only known from North America until this was done. Here's a new species that was on a rock about that big, covering nearly the whole rock. Looks like that when you look up close, so very distinctive, and again, so far only known in Britain. This has been compared with materials further in Scandinavia, and you'd think we probably share a subset of what the Scandinavian diversity is, but in fact, we've got a lot of unique diversity. So we're finding things that are new all the time up there, and there's a lot of work to be done. Surprising. So let's just think about scaling that up. So from well-identified herbarium material to a big, ambitious initiative called the International Barcode of Life Bioscan Project. The idea of that was to take specimens, millions of specimens, generate hundreds of thousands of barcodes from them, scale it up to start thinking about species interactions, so things that you actually can't see, but we can still generate sequences from the symbionts of other things. And then the next phase, this is the really aspirational phase, is the planetary biodiversity mission. The idea of that is to take tens of thousands of bulk samples, that's air and water and soil, and use barcoding as a baseline to figure out what we've got and how is it changing. Well, I would say that if we're going to be doing this, looking for changes over time, we better have a pretty well-populated reference database. So these barcoding initiatives across the world that are happening and have been happening for decades in fungi are really important. And it will allow us to start to unpick this trend to some extent, which has so far only been established using 5,000 animals with backbones. So do we have these same kinds of trends with other forms of life like fungi? Open question. Okay, so my last story is the future, looking forward to the future and thinking about biodiversity management. So here we have a map of the rainfall patterns across Great Britain and Ireland, and the dark blue is areas with a lot of rain. And when I mean a lot of rain, I mean a lot of rain. So that is 10 feet of rain on the West Coast. That's a lot of rain. And that promotes the development of temperate rainforests. Temperate rainforest is a whole different thing than the tropical rainforest. My kids go to school in Scotland and they come home, Mom, I heard all about rainforests today. Oh, you heard about the temperate rainforest? No, the tropical rainforest with the monkeys and stuff. I'm like, we have it here, but they don't talk about it. It's a very underappreciated kind of habitat. Like tropical rainforest, high humidity, we've got year-round mild temperatures, but for the temperate rainforests, they're globally rare. Fewer than 1%, less than 1% of global land surface is covered by temperate rainforests. And they don't have at all exceptionally rich plant diversity. So we've got 5 or 6 species of trees, that's about it. But in a single small fragment of a few hectares, you can have 200 bryophytes, 200 or 300 lichens. So the epiphyte diversity is ridiculously high. And not only is it ridiculously high, but it's of really high conservation priority. So they're globally rare habitats, and in the west coast of the UK, we have the most intact parts and fragments of these habitats that exist in Europe, certainly. So they're high conservation priority, but unfortunately they're also highly fragmented, so there's little pockets of it left. There's new initiatives to be trying to piece these things back together and promote connectivity and plant trees, but how do we do that in this globally changing landscape of temperature and pollution when we really don't know how these species work? So we had a really exciting project that was funded by the Scottish Nature Conservation Organization. It was a PhD project by Sally, and the idea was to try to understand something about the resilience of these temperate rainforests. So she built landscape scale dispersal models and did that by doing bulk sampling. So this is a battery pack, this is a motor, and on the top of the stick are some whirling arms with Vaseline on them. So this is a way of collecting spores. And this was an array, maybe you can see there's some here, 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 and they step out up to 20 meters away from the target tree. And those arrays were deployed over and over again so that we have a really good picture of how frequently individual target species were captured at what distance. And using lots of other survey data from the sites, including the frequency of colonization of the target species, the frequency of extinction of those target species, the distribution of the trees in the landscape, she was able to build this amazing model of just what kind of colonization probabilities you'd expect at different distances. So this is the one for this species. She studied 8 different target species. The graphs are all pretty similar. So this graph shows time on the x-axis up to 200 years down here, and the number of trees colonized up to 50 on the y-axis. And the different colors of these series of points have to do with the distance from the target tree where the colonization is predicted to happen. At 1 meter from the target tree, it takes about 25 years for 5 trees to be colonized. Here is, that's 60 years for 30 trees to be colonized, and it peaks out at about 45 trees after 200 years. At 20 meters and 50 meters, the picture is fairly similar. There's a bit of a delay. It takes 80 years to reach the peak. But at over 100 meters distance, the colonization is really, really low. These things just have trouble getting around, really big trouble getting around. And that's important because if we're going to try to be re-establishing and connecting these patches of forest together, we need to know how these species are going to respond to that. So it's really exciting. The managers of these properties are super excited to have data to help them understand how to plant trees and where to plant trees and what species of trees and how far away they have to be to promote the diversity of these epiphytes, which make them special in the first place. So the next steps then, so this is the forward-looking part of this, is to be using these systems to be ground truthing the bulk sampling method. So we've got, we've done that. We know that we can sample spores. We can amplify up the DNA out of those things. So we're going to be looking for doing some ground truthing against inventory-based data. So we've got some places that have been sampled really well. We've gone out. We've got lots of collections. And now, can we pick them back up? Can we see the signatures of differences across these different blocks of forest types using the bulk sampling? And I think one of the things that's exciting about this is in the UK, and I'm sure it's happening here too, is that the idea of using bulk sampling has just totally taken off. Everyone's really excited about it because we know there's a massive capacity gap. Kathy was talking about how few people there are who can do this work. So hey, well, there's a lot of people who can just generate some DNA sequences and do the bioinformatics of getting those identifications back out. But actually without any kind of good baseline, so the reference database is being well populated for the first thing, but second of all, without having best practice for understanding how sensitive your method is, what are you catching and what are you not catching? Does dispersal kind of method have anything to do with what you capture and what you don't? What are the frequency of the species in the landscape, and can you pick them up? So all of that is open. We have unknowns, lots of unknowns for this. And people are out there selling you their 250 pounds of sample. Oh, I can tell you about all that. But it's a giant black box. And I think it's really important to start getting to the root of that and understanding exactly what we can and can't say using these methods. So I hope I've demonstrated then that using collections for conservation baseline works. We can evidence losses. We can contextualize current trends. We can link experts to non-expert users. We can discover new species, and we can even answer management questions. So it's really exciting times. At the Botanic Garden, we're lucky to have 4 different gardens. So we've got a lichen trail for every one of those 4 botanic gardens. Yes. And we have high school students coming through. So we have about 1 million people coming through the doors every year at the Botanic Gardens. And it's a real opportunity for engagement and education. We've got lots of different education things happening, which I think is another important piece of the picture, obviously. So the more people we can inspire and get involved, we can build up our capacity. So that's what we're hoping to do. So just a few of the funders who I highlighted here in those projects, and an open invitation for all of you to come and use our collections. Thank you very much. So I think I have time for questions. Anybody has any?
Questions over here.
Yes, there's one in the front.
I'm wondering what the error bars would be on those loss estimates.
The error bars on the loss estimates. I mean, it's not a massive data set. So we had 100 species that we found across that study. And so that study, we had to limit it to places where we had the dates really, really well established. So you can date these timber frame buildings based on their carpentry. So they have different joins for different kind of even 10-year periods. It's quite amazing. So we were but because those dates are associated with only the big timbers, we had to limit the data just to the timber species. So then our losses there, that's 56 species in total for that data set. So it's not exactly a massive data set. But I would be surprised if those estimates are very far out. So because we have a huge amount of understanding of how sensitive these species are, especially to sulfur dioxide. You know, they've been used, there's an index that was published and was used for a long time when that was the main pollutant. So I don't have any reservations of thinking of those numbers as pretty realistic, actually. So in the 1970s, there was a fantastic survey that was put together by a guy who was a high school teacher and a lichenologist. He sent surveys to 10,000 school children. They went out to count the lichens in their schoolyards. And across massive swathes of lowland England, there were 0 or 1 lichens. And that 1 lichen was something we don't even find in the landscape anymore because it's a real acidophile. It needs loads of acid. So looking at the changes from having no lichens to having lichens in central London, we know the changes are real. And the recovery is now happening, but in a new pollution landscape with lots of nitrogen. So error bars, yes, there will be error bars. I don't know the size of them, but I believe the magnitude of the differences. I hope that answers your question. Anybody else? Everybody's hungry. Thank you for staying with me before lunchtime, so thanks.
Okay, a couple of announcements before we break for lunch. First of all, we do have lunch, and then following that, if you signed up for the glass flower tours, and the speakers are, you get to have a special tour of the Farlow. And I also wanted to just take a minute to thank Jeanette, Dawn, and Michaela. I have some little things for you. Jeanette, a book, and a fungal chocolate bar. And Don was the director of the Herbaria for many years, the director of the Farlow, and the curator of the Farlow. He's been a really important mentor. He's keeping everything going. And if you have already read that book, then I'm going to change it with something else. And I don't know, Michaela, if you're still here, but I have the Lichenpedia book for you and some chocolate. Thank you, everyone. I just loved all of your talks, and I just so look forward to the conversations we have this afternoon.